Welcome to Recombination Nation. Today, I have exactly the kind of guest I've been hoping to get on the show for, well, since I started a year and a bit ago. Sebastian Coccioba is an independent plant biotechnologist who has set up his own home laboratory and is doing all sorts of amazing high-level techniques that most people wouldn't guess that you can do on your own with a relatively simple and accessible setup with a bit of uh, research and determination. And apart from that, he has also been out there advocating to encourage other people to go down a similar path, which I absolutely love. So that's my little snapshot, Sebastian, of how I see you. Do you want to tell us a bit more about your journey and what led you to setting up a laboratory in your own house?
Oh boy. Okay, where to begin? I think I guess at the very beginning. So, um I've been interested in the concept of science since I first heard about it in like middle school when they were telling us about like the scientific method and like how people learn things. Because a really early question I had was how do you know, right? Like how do you actually how do you generate knowledge, right? It was far less articulate back then, but it was akin to how can we know when nobody knows?
Mm.
Because throughout my entire childhood and adolescence, people were telling me things. They're telling me knowledge that they had, but I never really had a connection to how knowledge is generated at the very edge of our understanding. And that took an awful long time because at first I was kind of enamored by the aesthetic of science, right? The idea of just like exploring the unknown and things like that, right? Just very Star Trekky, but grounded. But I didn't really have the ability to be like a profound academic by any stretch of the imagination. Like I was a B minus student at best, right? Like my uh, my academics was meh, but my curiosity was very, very high. And so I kind of fell in love with the experimentalist aspect of science. Like every time a science fair came around, we had it once a year. I loved it. I would just pour all my time, neglecting my other subjects, into making this thing happen. And yeah, I was just like super, super fascinated by it. Like Bill Nye and Carl Sagan were big heroes of mine growing up. I watched Cosmos a million times, and learning from Carl Sagan that science is a way of thinking didn't really resonate with me until much, much later on in life. I didn't quite understand what that meant. And so life happened. I got into a state university, did that for about 3 years. And then because of family and financial reasons, I had to drop out. So I had to completely pull out of university and I was unable to continue. But while I was there, I happened to work at one of the only plant labs at the university because I just didn't want to do medicine or cancer research. It was like a medical adjacent state school. I didn't really feel like that was my calling. I was pre-med at first. I was like, I want to be a doctor, and then I'm just like, I don't have the grades, so I had to back down from that path and decided to try something else. And so I stumbled upon that plant biology lab because in order to graduate from a major in biology at that university, you had to do a research rotation, right? And so I got a chance to work under Vitaly Satovsky, which, unbeknownst to me at the time, is one of the godfathers of agrobacterium, one of the main researchers that understood how genes are transferred from this microbe into plants and how genetic engineers use that to transfer their DNA of interest into plants. So he was, while everybody was making things with this technology, he was trying to understand the underlying mechanism that enables this technology. Like how does DNA transfer across domains of life from bacteria to eukarya into
Mm.
plants?
Mm.
And so that wasn't really it didn't it's not that it didn't inspire or impress me, but I just didn't know the magnitude of how important his role was in plant biology until I was well away from university life. But while I was there, I kind of got bit by the research bug, like really badly. It was such an addicting process. And I told him, I'm like, I don't have good grades. I'm not the best student. He's like, but you're passionate and you're interested. Because at around the same time, I was learning about orchids and how to propagate them. Because I wanted to give my mom like a really big Mother's Day gift. And orchids are super expensive. They're like $60 a piece in most places. But every Wednesday, this large housing and construction like warehouse store, Home Depot, would throw out their orchids that weren't flowering. And so I would like collect them from the side of the street and bring them home. And I was like online or early kind of search engine internet era. And I was just asking, like, how can I make more of this? And I
mm,
stumbled upon this micropropagation community of people like orchid enthusiasts or
mm,
flaskers, as they call themselves, where people would like take small cuttings of orchids and propagate them in vitro. And so it was such like a burgeoning, I mean, such a blossoming field full of people who were like super interested in all aspects of plants, but not the engineering side. I didn't even know you could engineer it until I went to that university lab.
mm,
Um, but in order to pay for some of the from some of school, I actually started a small micropropagation racket in my bedroom, right? Essentially, like the beginnings of a formal laboratory. And I was doing flasks on eBay. Like people
mm,
would send me their seeds, I would give them back 2 milk jars full of germinated seeds,
mm,
and that was kind of like a service I was doing. So I was learning tissue culture kind of indirectly.
mm.
And so when he asked me why should he pick me for this thing, I'm like, I know how to do tissue culture. And he immediately slammed the brakes. He's like, what? An undergraduate that knows tissue culture? I'm like,
Yeah,
yeah, it's super interesting. And I showed him some pictures. He's like, okay, you start tomorrow. You
yeah.
should start today, but you'll start tomorrow. And so he kind of took me under his wing. And so plant biology is like notoriously underfunded. And so one of the most formative moments was actually in learning how to use alternative materials to do the same science.
Hmm.
So across from our department in the biochem division, people had like Eppendorf branded lab coats. Everything was super shiny and schmick, like multi-million dollars worth of funding.
Mm,
And we on the other side were using like Jello cups and toothpicks as inoculation loops, like trying to keep things as cheap as possible.
mm,
We were like a ragtag motley crew of researchers. But he kind of taught me that resourcefulness that then resonated throughout my entire career, and I owe him a huge debt for that.
mm,
Um, and the when I told him that I had to drop out, he gave me a gift of a small piece of agrobacterium, a small tube of it, and said, like go in peace. Like I know
mm.
you want to continue this, and biosafety level 1 is not regulated in the United States, and so there is technically this weird gray area where you can do this at home.
Yeah,
Not every
yeah.
country allows you to do that, but that was such an enabling point for me because then I could finally continue all the things that I wanted to do in his lab, but at my place.
Yeah, yeah.
But I started looking around my lab and I'm like, okay, I'm missing so many things that I got used to. And so that took a couple of years to kind of build up the material side of things, right? And at first I was a generalist because I'm like, I need this and that and that, and I was working odd jobs to pay for it. And then I ended up using like maybe 20% of what I had, right, to be a little bit more focused. So that was a hard lesson on being question focused rather than being a generalist, like
Hmm.
capable of many things.
Mm,
Yeah, and so fast forward a couple more years, and I wouldn't shut up about what I do on social media. And I kept on like posting my adventures and trying to do genetic engineering. And I was working at the School of Visual Arts at the time, at a bioart laboratory made by artists for artists. And I was kind of like a scientific advisor in that space. And one day, through like some conversations, we were talking about the color blue in nature and why blue is super rare. And my boss was like, "Can we make a blue rose?" And I'm just like, Maybe," right? In that naivete of just like, "Yeah, I guess so," right? And so that put me on this like journey the to like it was kind of like the rumblings of flower design.
mm,
I didn't know I wanted to be a flower designer at the time. I just found it such a fascinating medium to kind of explore. And I drank a little bit of the art Kool-Aid while I was there, so I took a lot of the sentimentality and the deconstruction, that kind of postmodern vibe of art education. I was getting kind of like an indirect art education at the time
mm,
by working with these students, and that really made me. It gave me some really interesting levels of ambition that I wanted to see if I can somehow turn this into a medium.
mm,
Right? Can I make art using flowers as the physical medium, right?
mm, mm,
And that question became from like a hobby thing on the side to full force, like this is what I want to do with my life, right? And slowly but surely through social media research contracts, I started like paying the bills using my hobby, where scientists and academics would ask me how if I can do something for them like really quick because they see that I'm doing it, right? So the
mm.
proof is in the doing. I didn't
Yeah.
need a CV or an education formally. If I can do it, then I can do the same thing for them, right?
Mm,
And so I became this one-man CRO, this contract research organization to pay the bills, and then I moonlight at night as an artist to try to
mm.
generate flowers that people haven't seen before and use that as a medium for expressing the human condition.
I've got so much I want to talk to you about, but firstly, before I
Yeah.
forget that you called your tissue culture early lab a racket, I
Yeah,
love that. But I want to just briefly touch on the idea of theoreticians versus experimentalists in
sure.
science. And maybe, do you agree that today formal academia is almost completely dominated by theoreticians? It's the people who are really good at doing exams, filling out forms, writing grants, but you put them in the lab and you make them get their hands dirty and they're like, oh, and they kind of they don't have the passion and the drive for it. And I wonder if that's why your professor, when they saw that you already had the hands-on touch for doing plant tissue culture, they're probably used to people coming with perfect grades, and after 3 years, they're still pretty crap at it. Sometimes they
Yeah.
probably never get it.
Yeah, there's yeah, I mean, what's unfortunate, and I forget which one of my friends told me this, and it resonated with me deeply that the profession of science, the job of being a scientist, often gets in the way of the vocation of scientist, right?
Mm,
Of like, there's the kind of romantics of teasing knowledge from the aether, if you will, right? Like that kind of gentleman science of the 1800s, ideally without the sexism and racism and all that jazz.
mm,
But that spirit of exploration for exploration's sake is still there in many of those academics. It's just in order for you for your job as a scientist to be maintained, this grant writing cycle and only doing things that are grantable, right? Doing things that could be seen by a study section as plausible to be
mm.
allotted money to is unfortunately limiting, but I don't think it's intentional. I don't think that it's the spirit of science is gone. It's just the mechanisms, the gears of academia kind of grind that out of you,
Yeah,
right?
yeah.
And so if the ultimate goal of an academic is to become a tenured professor, then what your ultimate goal is you become a manager,
Mm,
right? You manage a lab, you write the grants, and then you have your curiosity flow through the able hands of your students, right? And in return, you kind of train the next generation of researchers in the process. But it is unfortunate that like I mean, there's always jokes of saying like, oh, the PIs in the lab, things are going to go bad, right? Because they've lost their touch more often than not
mm,
physically doing things.
mm,
But like the thinking is there, definitely there. Obviously, the writing is fantastic. The theory craft, like you said, which is at the heart of what science is. The final science output is the communication of
mm.
natural observation of natural phenomena,
Yeah.
and uh, and that still exists. And I mean, there are many PIs that I've worked with that are very hands-on, that are actively developing methods, especially like methods laboratories that are focused on developing method. All
Mm,
the theory craft is still directly applied, right?
mm,
Um, yeah, and just, I mean, as an outsider who's never been an academic by any stretch of the imagination, but have collaborated with several academics, I get to see kind of how the sausages are made. And I also got to see how it really drains you in many ways because there's so much university administrative work that you have to do on top of your teaching, on top of your research,
mm,
that kind of splits your brain in three directions. And while, it pays okay, and you have the at least the prestige of saying that you're a professor, and then and whatever little is allotted to you nowadays because of all the funding issues, you still have to serve the university at the same time. And
mm,
so much of this like bureaucratic stuff, this red tape and all of the things that they have to work through limits the ability for them to like creatively express themselves. So
mm.
like they rely on the writing. Many of my professor friends rely on the writing as their vehicle for scientific expression if this were a craft,
Yeah, yeah.
because everything else they don't have time for. And so there's a bit of like a bittersweet melancholy about the higher up, the more advanced you are in your career, the less science you do,
Yeah.
right? And the more
And the
science
fewer risks
management
you
you
can
do.
take sometimes, particularly in the
Yeah,
middle, the middle part of the career where you should be swinging for the what do they call it? Swinging for the
the
nets?
fences,
For the,
the
swinging for the
fences.
fences. Yeah, we don't play
Yeah,
baseball in Australia.
that's
Yeah,
right.
your hands are kind of tied for taking those big risks because if they don't pay off, your career can basically be over before it really gets to that final
Yeah,
goal. Uh,
yeah,
well,
and
well, I wanted to
it's
ask,
go
do you
ahead.
think as an outsider you have some advantages over academic or industry-based researchers that you
Like
get to follow your passion and take chances?
I do, but there's always a cost, right? And so like there are three things. You have budget, you have resources, and you have questions, but
Mm,
you have to pick two,
mm-hmm,
right? So it's I have total creative freedom if I'm at the helm of my ship. And I can choose what research to do. Now, assuming that I have the time to do so, because right now a lot of my time is gobbled up by client work to pay the bills.
Mm,
A lot of family kind of catastrophes happened over the last couple of years, and I inherited a house with a pretty hefty mortgage all of a sudden. And
mm,
so, like, a lot of my work that I would have been a bit more free for, let's say paying a lower rent, is now kind of tied up in this very old house that I'm repairing.
mm,
And so, like, life makes me not have that much time. But when I do have time, it is the most liberating, like, freedom I have. Like, it's if you're a musician and you're good at a musical instrument and you have like an hour to just doodle for yourself, there's
mm.
just a feeling you can't describe,
Yeah,
right?
yeah,
This like competent eloquence at a craft, and you just do it for yourself,
yeah,
right?
yeah.
And so, what I try to do to maximize the amount of time I have in working for myself and for the projects that I love is to take on projects that overlap heavily with my interests
Hmm.
or push me to the edge of my competence. So at least I'm learning,
Yeah,
right? I'm kind of
yeah,
like outsourcing what grad school would have been by taking on projects that are at the edge of my understanding.
yeah,
Like I know
yeah,
I'm competent in executing, but let's say I've never done that one before,
yeah,
right?
yeah.
Because I'll never overpromise and say, "Oh, I'm going to do this thing I've never done." I'd be honest with them. Be like,
Mm,
I have the capacity and I have enough experience to be agile at learning something new, but it is something new for me. And luckily, the people who've reached out, because it's always been by word of mouth, have been heavily overlapped because they only reach out when they see me doing something that's akin to what they want,
mm,
right? Or within their wheelhouse.
yeah.
But the few times that I did collaborate directly with academics on just a purely academic paper, like making, like being a scientist and publishing, it felt kind of like therapy for them because they got a chance to kind of forget about the grants and all the things and just do a cool project together, right? So for me, as an outsider, I see, sounds a little cheesy, but I do see science like jazz, right? This kind of like democratic way of expressing yourself. And whenever a friend of mine who happens to be a professor drops a new paper, it's like a new album drop of my friend's band, and I
Mm,
can't wait to listen to it.
yeah.
And I give like immediate feedback, and just I'm genuinely excited about it. Like I'm a fanboy for so many of my academic friends. But and it's cool. I mean, they even told me is like I've never had somebody ask for my autograph. Like I got an autograph from a couple of my science heroes, and I printed out their dissertation. I'm just like, can you sign this? Right? I mean, for no ulterior motives. I was genuinely interested in them as craftsmen and what I can learn from them. Right? And just like looking up to the fact that they're capable of doing this. Like good, anybody can be a good scientist. Great scientists require a lot of luck and then just like a specific meh that I can't describe.
Mm,
That I always admire because when you sift away the luck, you get to this like perseverance, this like stubborn, like thick-headed stubbornness of saying, no, I'm going to push through all this crap because I really, really think that this makes sense. And those stories are so inspiring because so much of science is unknown,
mm.
right? Like people say
Particularly
like, oh,
biology.
oh my God, so
Yeah.
much, so much. I mean, there's like industrially viable catalysts on your shoe right now that no one's gonna know. So you're gonna walk out the door, right?
Yeah,
Like,
yeah.
but in that unknown, we're all fumbling around in the dark, and you feel the level of acceleration of competence towards almost equal happens fairly quickly,
Mm,
such that I do feel to some extent my peers and I are equal in fumbling together.
mm,
Right? Like they have more experience because they've been formally trained, and my knowledge base is a little bit more Swiss cheese granular. Like there's going to be parts that I'm missing,
mm.
but at the same level, I can work with them as peers on a project, and it feels equally like we both don't know what's happening. Like we
I,
both don't
I
know where this is going.
I would almost guess, from how I've seen a lot of big famous laboratories work, that a PhD, for example, often gets trained up in one particular technique to be really good at it, and then they become part of a production line, but they come out the other end without broad skills to like manage the whole process themselves. I think you're in compared to some like people with a PhD, you probably have advantages that you have been forced and you've had the opportunity to learn a little bit of everything.
Yeah, yeah. Like being in survival mode got me into a generalist state where I can
Mm,
take on most projects within my wheelhouse, within BSL-1, and
mm,
that gives me a sense of advantage to some extent. But again, like at the end of the day, if this wasn't my job, I would do something else and I would just do this for pleasure anyway. Like I do
Mm,
science for the love of the game. That's why I call myself an amateur.
mm,
And people often conflate amateur with novice, which are two very
mm,
different concepts.
mm,
Like novice, you're just started. Amateur just means you love doing it, but you can be
mm,
incredibly skilled and love doing it. You can be
mm.
an accomplished painter without being a professional painter. The
Yeah,
same thing.
yeah.
But within that scope, there are advantages that I've learned enough to be able to tackle most of the problems within my sector,
Mm,
right? To be able to like interface with those problems, not to solve them. I'm no Mozart by no means, but just to interface with them and be a competent biotechnician, right?
mm.
And even fledgling biologist, like my biology theory craft is dismal, but I'm getting there. Um, uh, you're a lifelong student of evolution. Like there's no biological Mozarts, right? Nobody's born with the gift of understanding the living world because we barely understand it as a collective, right?
Well, on
Um,
on on that
yeah.
note, do you think there might be some advantages to not receiving all of the formal theory training? That some of that theory is probably not quite right, and sometimes outsiders wander into a field completely, ignorant of the established way of looking at things, and they spot that there's better ways of doing things. I wonder if you and more broadly independent biology researchers in this century could be a source of those kind of ideas that academia
Yeah,
has become too rigid and too formalized to spot as easily.
yeah, I think both in academia in navigating like interactions with academics and in industry, I do find that I can approach a problem more creatively because I'm pulling in all of my experiences from different places at the same time. But it's there's this deep irony. It's almost always a double-edged sword,
Mm,
right? Like I'm coming at it from I'm complete like I don't have the tunnel vision to know what's not possible, but then at the same time, I could also be spouting things that are inherently impossible, thus wasting time, right?
mm,
So
mm.
like Carl Sagan was just like, yeah, what's it called? They all laughed at this one brilliant researcher, right? Until he was proven right or something. But then he said people also laughed at Bozo the clown,
Yeah,
right? Like
yeah.
laughing and mocking for being an outsider isn't always a path towards like disrupting a current space in a
Mm,
positive way. Like sometimes your lack of competence or lack of knowledge is actually a detriment,
yeah,
right?
yeah,
So I think what's important is as a researcher as an independent researcher is to find your research question and really go as deep as you can with your interest, right? Like narrow it down to a singular question and dive into that and give yourself that PhD level specialization such that you can hone deep thinking,
mm,
right? And because deep thinking is so hard to describe and so difficult of a skill to generate, but that only comes about by just constantly thinking of this one thing for a long time. And it's this
mm,
meditative state that trains your brain to be able to assimilate large amounts of data in a very, very deep way.
mm,
And that tends to more often than not come from being a hyper specialist as a PhD in a PhD program, right?
mm.
But the caveat is the deeper you think, the less broad you can think,
Yeah,
right? So yeah, I see it as just those graphs that have like a polygon of different points, and they're
yeah,
just like competence, speed, eloquence, creativity, and they're just like all over the place. This type of archetype, right? Like no one person can be both broad and deep at the same time,
yeah,
right?
yeah.
So I mean, I'm still reckoning with that of just going. I have interests that were very broad at first because I didn't know how to think deeply. Now that I'm practicing to think deeper, my interests are actually narrowing
Mm,
because I'm finding more fulfillment and more joy from diving into a smaller amount of questions than really broad questions all over the place. It was a focusing of attention
yeah,
that I think if I went through grad school would have taught me that skill, but then I would have been forced to focus on the attention of the lab that I had the fortune of joining, right?
mm, mm,
Because most people unfortunately don't have the full flexibility of fleshing out their thesis as they want it to be. And to be frank, at around the age of a PhD student, they tend to not even know what they would want,
yeah,
right? You do a PhD rotation and you'd see different labs and see what they're doing and kind of suss out the vibe. But you almost always know what you're interested in hindsight, which is unfortunate.
mm, mm,
Because I mean, it took me a really long time to understand what my like core questions are, right? And like for me, it's change, right? So I'm really interested in change.
Mm,
I'm really interested in mutagenesis. I'm interested in like the longevity of change over time, especially man-made change. Like if you make a transgenic plant, how long will that gene that you added, that transgene, how long will it last in a population,
mm,
right? How long until life kind of makes it fade, or go away? And more importantly, how can you engineer traits to be long-lived? How can you engineer traits for geologic time? Because so many of
interesting.
our genetically engineered plants that are released legally today will probably fail very quickly, especially crop plants like corn. Like
Mm,
you put those seeds in the wild and they're so hungry. They're on life support all the time with all the nitrogen we're feeding it. And so like leaning into the tenacity of life, how can we make tenacious transgenes? Right? Those are kind of like the
interesting.
questions that I'm really, really interested now. But it took a really long time of just like trying a whole bunch of crap out to figure out like what my vibe is. Like what is the style of my music if I'm a jazz musician? Yeah.
You're very much aligned with the landrace crop breeders that I generally talk to, who are doing all sorts of wide and crazy hybridizations between orphan or novel crops, and like trying to
Hmm.
replicate that traditional path of biotechnology that's like, thousands of years ago. That's where the
Yeah.
original crops came from, usually through very unlikely hybrids, and then humans running with them and turning them into something useful and persistent. But let's dive into some of your specific projects. You have quite a few on the go, but the one that
Yeah.
grabbed my attention first was this white petunia that you stumbled across that you want to turn into a model organism.
Yeah.
You don't see many new model organisms being introduced into circulation. So
Yeah.
tell me more about that.
Sure, sure. So when I settled on I want to be a flower designer, right? I started like taking the concept of the artist side of things fairly literally, right? And I would be like, okay, if I want to be a designer, what is my canvas? What is my clay, right? Like what am I going to use? Because all of my experience with genetic engineering showed that so much engineering is focused on the chassis, the thing that is receiving your gene, just as much as the genes themselves. Like currently, we don't have technologies that are chassis agnostic, where you can make a transgene and it'll just work in any land plant, right? It's often tailored, it's often hyper specific. Sometimes because of regulations, it has to be cisgenic, meaning you use genes from inside the genome or the genus of the organism and borrow from there.
Mm,
And so um, there's a lot of pressure to focus on a singular point, right? And I said, okay, well, an artist can be a sculptor and a painter and a musician, sure, but a lot of accomplished artists tend to be singular in their focus, right? Like Renoir and Monet, right? Like you paint, they just painted,
mm,
and so so I'm just like, okay, what is my canvas? And so for a whole decade, I've been looking into trying different plants as my canvas. So I cut my teeth on tobacco. That's like my bread and butter plant. It's super amenable to newbies for making mistakes and whatnot.
mm,
But its flowers were kind of uninteresting for me. Like when I think of flowers, I think of something that's really elaborate, flamboyant, expressive, and a grand departure from the rest of the plant. And in tobacco, the leaves were enormous, and then the flowers are these itty bitty little trumpets, right?
mm,
And the petals weren't even separated. I quickly became like a bit snobbish when it came to flowers. I'm like, I don't really like that. That's not interesting.
mm,
And um, and then it dawned on me in that process that I definitely need a white plant, right? Because I need a blank canvas. Because in the unlikely event that I'm interested in color, I'd want to have something that's neutral, right? And I want to be able to test circuits that might be poorly designed by me, but still working. And if I
mm,
have a white background and my transgene is a color like green, I can see that subtle green on the white petals than if it were into a leaf,
mm,
right? So I get a little bit more feedback. Okay, so I settled on white flower. Cool. Moving on from there, I tried every white variety of like every plant you could think of that's like commercially available, that's open heirloom pollinated, because I didn't want to step on any patents.
mm,
But I wasn't interested in Arabidopsis, right? That's like the flower model for plant biology. The flowers are just too tiny for me,
mm,
right? Um, my friend Nick Desnoyer, shout outs to another flower designer. He focuses on Arabidopsis and he's done a lot of breeding and CRISPR work to make the flowers larger, right? To make a rose out of what would normally be just four petals,
mm,
right? And so there's a significant amount of work in trying to make them larger. And on my end, I just wanted something where the majority of the plant was a flower, ideally, right? So I
mm,
started formulating this like idealized checklist easy to propagate, not toxic to animals, because I had pets at the time. Um, Large flowers, easy to tissue culture, transformable, ideally sequenced, but if not, maybe just a genome that's not a bajillion base pairs, right? Something that I could sequence myself
Mhm.
to some extent. And tried and tried and tried, and there was always recalcitrance, like the plants just would not respond to tissue culture. Or there's one caveat, like it takes forever to vernalize. It takes like a month in the fridge to simulate winter, right?
Mm.
Or they just don't produce enough seeds. There are just so many caveats with all the plants I tried. And then one day, one summer, my mom was at like a roadside flower stall, right, just trying to pick up some flowers on the way back, and she saw this like handwritten sign that said white petunias $5, and it was just like a tray of them. And she got it for me as a joke. Because early on, I tried petunias, white petunias, so many white petunias, and they were
Mm.
all terribly recalcitrant and awful, and some of them were patented, and I couldn't work with those. And she's like, just give it a shot. You haven't done it in a while. Just give it a shot. Right. So my mom, for the listeners, my mom is like my lab manager at this point, right? And when I do have a decent amount of client work and it pays well, I actually hire her full time as for it. I can pay her for salary and whatnot. And she like yells at me and tells me I'm running out of tips and whatnot. Just kind of helps me manage the lab day to day. But she also runs her experiments. So it's a nice kind of like it's like having a postdoc in the lab, right? While I'm the
Wow.
PI. Um, and she has no science training, and so I taught her the way I taught myself, and she has her own little experiments. It's cool. But yeah, so she got it for me as kind of a prank, right? Of just going like, "You want to try petunia again?"
Mm.
And so I said, "Okay, fine. For you, I'll do it." And because it was kind of like a bonding moment. We both surface sterilized the leaves and whatnot. And I was too lazy to make like a petunia specific media, and so I took my usual tobacco stuff that I use for clients. I bleached the leaves. I made some leaf discs. I put it on a media. I said, "Okay, we'll see you in 2 weeks." And lo and behold, it started making large shoots within those 2 weeks. And it was the fastest regenerable plant I've ever worked with, like ever, hands down. And I was just in awe because I was working with tobacco media. So, like, it's super, super well understood, very, very affordable. And I have so much of it in my fridge pre-made. It flowered 25 days later. I even had flowers in vitro, right? Like it was
Mm.
just so amenable to all the things I needed. And because of the way in which I grow my plants in a hydroponic setting in really small tubes, the flower actually occupied the bulk of the total mass of the plant. It was
Wow.
just big flower and then itty bitty leaves. And I'm like, this is perfect. This is literally perfect.
Mm.
And out of just like riding that high, I said, okay, what if I try to infiltrate the petals? So as a way to test genes ahead of like trying to stably integrate it into the genome, which takes a couple months, what you can do is take the agrobacterium armed with your gene of interest and physically inject it in between the cell layers of leaves,
Mm.
right? It like spreads inside, the bacteria spreads inside, and then it transfers DNA into those cells that it touches. And ideally, you have a visual reporter, so it'll like glow green. And the
Mm.
brighter it is, the more powerful the expression, for example. Um, but I thought instead of the leaves, could I inject the petals, right? And so I prepared the same experiment and I injected with like a needleless syringe into the petals. And lo and behold, it flooded the petal beautifully. The entire lower petal limb was covered in agrobacterium on the inside.
Mm.
And 3 days later, I got a beautiful ruby red signal. And
Mm.
I'm like, this is it. This is my model. This is what I'm going to sink the rest of my life into,
Is
right?
this the Henrietta Lacks of plants?
So I don't know how fast comparatively, because there's so many things at play, right? Like I don't know. I don't think I can beat Arabidopsis in terms of convenience because
Mm.
what I have to do with tissue culture, people in Arabidopsis land, they just take the flowers, the flowering plant, and they dip it in the agrobacterium while the flowers are in bloom.
Mm.
And then the agro transforms the ovules, so when it self-pollinates, you make transgenic seeds.
Mm.
So you remove a lot of the technique from the equation, right? It makes it super, super accessible and it does cycle very fast. So I can't beat them on the cycling time. The iteration time is just gobstopping. Like my biggest envy with Nick's work is that he can iterate very quickly,
Mm.
right? If a circuit doesn't work, you can do it within 5 weeks, you get another result. For me, it takes 3 to 4 months to do
Mm.
stable transgenesis in my plant
Mm.
because I have to regenerate from little bits of tissue. But the benefit of that is that it's a big old flower, right? It's huge. It has a lot of real estate, and more importantly, you're starting with a really simple shape. And so as a result, any modifications to the body plan genes, the homeotic genes, could be more pronounced. Because what I want to do, my goal here would be to take Petunia, and morphologically change it such that if you put it in front of like world-class botanists and taxonomists, they don't know where to key it out, right? They're like,
Yes.
where does this belong, right?
I love it.
And just like a grand departure, because part of the from the art side, part of my focus is departure, right?
Mm,
It's change, right? It's like how can we move away from the already selected ornamentals that kind of have this undercurrent eugenicsy vibe because somebody said that that's a perfect rose, and
mm.
now everybody's saying this is perfect, but we don't like independently arrive at the same conclusion that that's perfect. Like it's a taught exercise, and I'd like to explore the morphologies, the latent morphologies inside that organism, and see
Mm.
what I can tease out by just modulating either through traditional breeding, which I've come to fall in love with, or through gene editing and transgenesis. Yeah.
Have you had any interest from the petunia breeding industry? Because you regularly see really novel forms and colors from more traditional breeding turn up in the market under PBR. Surely they would be interested in completely new variations that then they can incorporate into traditional breeding programs to
Yeah,
prepare
yeah.
market-ready
And so
plants.
yeah, and so I've been in contact with one very large horticultural grower, and I'm under NDA, so I can't really
Yeah.
describe too much. But their interests were very much like amplify what we already have,
Mm,
right? Like make what we have more or different, but not too different, because we don't want to lose that performance. Because so much of the value that a lot of these annuals, especially in like the petunia space, is that hardiness, that like the pink wave petunia, right? That
mm,
entire wave series is just so hardy and so robust and just endlessly flowering and just like spreads like crazy.
mm,
That robustness is such a polygenic trait that the worry is that by modifying body plans and whatnot, you could be messing with that bread and butter hardiness,
mm,
right? That's at the core of their industry. And a lot of them are also a little bit, not a little bit, a lot of bit hesitant to engage in things like CRISPR because the patents are so contested,
mm.
right? So like the main CRISPR patents are going to expire in 2029 to 2032, right? And a lot of them are just holding on until that expires for them to have a clear path to market, like a
Yeah.
clear freedom to operate within that space.
Yeah.
And so there is like the burgeoning of interest with transgenesis, but on top of the fact that very few countries in the world would allow ornamental transgenics to be deregulated, like the
Mm,
US and Singapore are one of the few markets that are the most amenable
yeah.
to bringing a new crop there. And as of recently, it's been a free process, like 15 months to be deregulated. And they care about mechanism of action. So
Mm,
instead of caring about how you put the gene in there, it's like, what does this gene do? And
mm,
do you have evidence that it's not going to make it weedy or spreading, et cetera, et cetera?
mm.
So bringing a plant to market in the US is possible, but it's still a headache. And then that CRISPR patent just kind of muddies everything.
Yeah. Yeah.
And so for the last actual couple months, I've been diving into irrational design, where I'm leading into like UVC mutagenesis as an example, right? Because I wanted to make change, but I don't want to get cancer. So what is like the lowest cancer changing thing? And it turns out that all of the chemicals that are mutagens, most of them are carcinogens, right?
Mm,
And I don't want to work with that in my home, especially, let alone in a formal lab. Like in my house, even more so, I don't want to work with it. And UVC is super readily available, and it does only a certain type of mutation, but in the presence of caffeine, weirdly enough, like lethal levels of caffeine that you should never drink, it causes very broad mutations.
ah.
And so I'm like leaning into this like sunburn and coffee, like sunburn and black coffee is like the safest possible mechanism. It's still toxic, it's still dangerous, but you can turn off a UV light. I
Yeah.
can't turn off a carcinogenic chemical,
Yeah.
right?
Yeah.
Um, but within that is just this like new layer that I'm extremely excited for. Like even if people are saying like, oh, you're only going to get TT dimers or only very specific mutations, I'm like, a mutation's a mutation.
Yeah.
And plants are mostly AT rich, so you're probably going to be hitting important things that they're two T's next to each other that fuse. So yeah, like a lot of my practice now has also been research necromancy, where I'm kind of bringing back old technologies that have fully expired the patents, but are still viable tech.
Mm,
Like I'm so in love with RNAi, RNA interference. All the major patents are expired, so it's kind of like public domain. Anybody can use it. And it's just so elegant. You just make a RNA sandwich with a little loop and it silences the gene, right?
mm,
But it can silence it in a gradient, so you get a little bit of silencing. So you get that kind of interstitium between totally off and totally on. right? There's just so much design abilities that you can do when you're not permanently modifying the genome, right?
Mm,
You're just momentarily interfering. You're kind of playing with the knobs rather than just switching on and off. And in that nuance and in that semi-irrationality of rolling the dice and seeing what happens, I found this like great fulfillment, this almost like a burden off my shoulders that instead of having everything perfectly precise or as precise as we would want it to be, just lean into the noise a little bit,
hmm.
lean into that irrationality and see if you can marry the casino with the intelligent design, right? With
Yeah.
the rational design. Yeah. And so far in my Snapdragons, I have a tricot that's stable, right? So it's producing three leaves per whorl at a time.
Mm.
I have a anthocyaninless mutant that just spawned from that UVC stuff. I have a whole bunch of freaks, and I can't wait to explore them even further. All of the changes I'm doing are so polygenic. Like one of them, the Snapdragon looks like a clamshell that smells like fake grapes. None of the Snapdragons smell like that. Just that one smells like methylanthranilate, like
Mm,
the smell of fake grape soda. And I mean, it's just such a joy to explore. And what I can do is take all my engineering knowledge and apply it to that one cultivar that already is freaky and add stuff to it. Add the things I know, the finger painting I do,
mm,
compared to nature's brushstrokes, right? And so, because I realize that there's a little bit of intellectual hubris to think that these changes that we're doing will only impact this one thing,
mm,
right? Because even when you put in a single transgene into a plant, where it lands in the genome is just as important, and it can impact greatly the expression. So you're still rolling the dice. It's just like more loaded dice. And so I would like to just do both and see where that takes me. Because if ultimately I want to express myself through the language of flowers, I don't have to be like German engineering about it. It's
mm.
okay to be a little Jackson Pollock from time to time, you know?
I love that.
And yeah, no, and I'm just madly in love with this concept now.
Well, this makes me think mutational breeding was really popular back in like the 40s and the 50s, you know, like the atomic era. They
Yeah,
were
atomic
blasting
gardening.
seeds with like every form of mutagen that you could get your hands on. But
Yeah.
do you feel that the trend in academia moved on prematurely? Like there was more to be developed using those techniques, but then there was some shiny new thing that came along that suddenly that's the only thing you could write papers about because it was the
It
next big
seems,
thing.
yeah, it seems like that's the case because there is something to be said about intellectually about the lack of control that random mutagenesis can give you, and also the amount of time, right? Because like there are species of bamboo that take 150 years to flower. So
Mm,
like good luck breeding that, right?
mm,
And anything that you breed, unless you cross it, it won't be heritable. It'll just be an epimutant that's momentary in that one organism, right?
mm,
And so there's um there's a lot there's obviously massive benefit to having precise control of the crops that you want and in a timely manner. But I do agree that I think the impetus to file grants, I mean to write grants for random mutagenesis has dropped in face of more precision things that require more infrastructure.
mm,
But if you still need a really high quality genome to find those targets. You still need to have freedom to operate with CRISPR. If you're an academic, it's fine. You can just do whatever.
mm,
But conceptually, you need so much more understanding of the underlying genetics of the organism to make great change versus random mutagenesis as a tool of understanding,
mm.
because a lot of sciences break things and see what happens when you break them, right? Like understand the working by deconstructing the non-working, right?
Yeah.
And crossing them in and seeing how these phenotypes carry. Like there's a tremendous benefit of the synergy of the two, which I find more troubling that people want to choose one or the other, right? Like traditional breeders would rather do mutagenesis than CRISPR, and then the academics would rather use CRISPR than mutagenesis, like mutation breeding.
Mm.
And I'm sitting here going, why not both,
Yeah.
right? Like why not use all the cheap sequencing to sequence the weirdos to understand what other polygenic targets we can hit, right? Like
Mm,
there's a way to apply great science to the randomness to better understand future precision edits. There's a feedback loop that we're kind of emitting through this process.
mm,
And I mean, I recently learned that there's a company in Japan that has a Cobalt 60 atomic gardening setup, and they can generate like 12 to 15 new cultivars of chrysanthemum per month, right?
mm.
Just
Wow.
through this process, just increasing the heterogeneity of these pools. Because you blast it with mutants, I mean, you blast it with a mutagen, but then you have to make it either self or cross it, and it's in its progeny will you see the segregation of those traits.
Mm,
And so some of them are very subtle in the parent in the M1 that was blasted, but then very, very specific in its in subsequent progeny. And so there is this kind of like waiting for Christmas for your present type of thing. It's like, okay, okay, I blasted it, but now I have to wait a full generation and hope that I didn't blast it hard enough that it's not viable or non-fertile,
Mm,
right? Um, yeah, no, we're definitely, I think now with AI at the helm to be able to process a lot of these polygenic traits and look for patterns that would take us forever, or maybe we would never look for it because we can't process all the things at once.
mm,
There could be a way to marry these things into a really synergistic process where you have that random mutagenesis that makes like miraculous species, and then you have this cheap sequencing to better understand it. Then you have RNA expression sequencing tied to AI that could help you kind of make a mental map of the networks required, the modifications required to make something look truly different,
yeah,
right?
yeah. Well,
Yeah, this now is like the best time to marry those two. Yeah, and I strongly recommend folks who are in traditional breeding to team up with an academic a precision crop engineer, and make some really cool things. I think the teamwork there is fantastic.
that's a question I'll ask in a second. Well, actually, no, I'll ask that now. Do you see, that's a really interesting idea. Do you see more potential for academic labs to collaborate with amateurs working independently? Because they're often
So
like really, they've got a hoard of like resources and goodies that they often like, they'll have $1 million machine that only operates like 1 day a week.
yeah,
And they also have a limit of manpower and flexibility to do really risky creative things.
yeah.
Is there a new opportunity for that collaboration to link
I,
those two groups?
I think there is, but it's starting to turn into like a was because like especially in the
Mm,
United States right now, the current funding landscape is dismal, right? And people are like shutting down labs left and right. They don't have enough money to pay their students. Anything that remotely says words like diversity, regardless of context, was flagged for it. I mean, it was a disaster. And as a
yeah.
result, um, this would have been a great question about 10 years ago, right? When we were still getting a significant amount of like the Biden biotech initiative pushing a lot of money into this. Not 10 years, like 5, 6 years ago would have been fine too, pre-COVID, basically.
Mm, mm,
COVID kind of ruined it for everything. But what I've been mulling over is if amateurs could get in contact with like large breeding houses, right? Because I think academics right now, unless you come with the prerequisite skills, unless you've done tissue culture in your bedroom, it's kind of hard to engage with those labs because they would also have to give up some of their resources to train you, right?
yeah.
On top of that. So you would have to come pre-trained in some capacity. And
Mm,
finding folks that have the strange desire to teach themselves at great personal sacrifice is a little bit rare, right? And so I mean I would love to see more folks through my path. I wouldn't recommend my path to anybody. Like it is
mm,
just it's fraught with adventure and misadventure, but also a good amount of luck that I was in
mm,
the right place at the right time to meet the right kind people that
mm,
could give me a shot. But for amateurs to reach out to large breeding houses that don't have the time and said, "Hey, I really love this one crop you have. I would love to do strange things to it, and then you can pick your litter, right?
mm,
You can pick the ones that you'd like, and then
mm,
I'll do weird stuff to it later." I think that there's like opportunities for amateur breeders to interface with these folks using all of these modern tools, using this kind of decoupled from academia, because academic research with industry partnerships tend to have like material transfer agreements and royalties, et cetera, that may not be conducive to anything but very large, clear benefit projects, right?
mm,
But the smaller things would be like, hey, I would love to do this if you just give me full access to your germplasm,
yeah.
right, of that particular cultivar. And then you can flesh out a deal or things like that. I think there are entrepreneurial opportunities for amateurs to be able to interface from like ornamentals, for example.
Mm,
But from academia, it's a lot of the times you need to generate a reputation in order for academics to treat you even remotely seriously. And a lot of that just takes a tremendous amount of time,
yeah.
right?
Yeah. It's a chicken
Like
and egg problem. Yeah. Yeah.
yeah, like for me, if I didn't shut up about what I've done for the last decade plus, I wouldn't be where I am today or who I am today.
Mm,
But at the same time, I spent a decade doing that,
yeah.
right? And I think you can do it faster if there was just like apprenticeship programs,
Mm,
right? Like biotech apprenticeships. But then you start flooding the market with folks that are expecting this type of partnerships rather than it be like serendipity,
Yeah.
right? Because once you expect it, there has to be pipelines, and then there has to be cost benefit analysis, and it becomes bureaucratic very quickly.
Yeah. Yeah.
And so, I mean, I would recommend any amateur to just like cold email an academic and go like, I'm super passionate and I want to do this. Would you at least guide me? Like,
Hmm.
don't ask for resources, ask for mentorship because that's
Yeah.
priceless,
Yeah. Yeah.
But within that space, I think there are many academics that would love to have these type of side projects, especially where they're not like solely responsible for the resources, the grants, et cetera, et cetera.
Yeah.
If you're coming in and be like, hey, I got a little lab, I want to do some stuff. Do you want to collaborate? I
Yeah.
got an idea.
Yeah.
And for me, the best output that academic can give me is that feedback of going just like, hey, just hear out my idea for a second and
Mm.
just pick at it, right? Just
Yeah.
a little bit. Like
Yeah.
informally, you can be as vicious as you want. If you don't have time, I totally understand. But if you can give me that feedback, because honestly, I've never had a mentor in this space and I've been craving it so much. But recently I got to the point where technically I'm competent as my academic collaborators. And as a result, it doesn't feel like a mentorship relationship. It
Yeah.
seems like two veterans in the trenches doing something as a collaboration. And it's
Yeah.
awesome
Yeah.
and I love it, but I still crave that education in the back of my mind.
Mm.
So I try to be a mentor for folks that cold email me going like, hey, I love what you're doing. How do I start? Right. And my
Well,
canned response always is start with a question
yeah, yeah. Well,
before.
along that note, I wanted
Yeah.
to ask you, like, you've been really advocating to encourage other people to follow this path if it's something that sparks their interest. And if someone wants to get going in this direction, um, where would you recommend people look for resources to build a simple home laboratory to get started?
So, my Bible growing up was a book called Plants from Test Tubes. It's now in its fourth edition, and like the magnum opus of my career would be to write the foreword of the next edition. That's how
Hmm.
much I love this book, right?
Mm.
It's from a gardener's perspective, everything necessary to do tissue culture and even like make a small operation for selling micropropagated plants. It teaches you how to make a laminar flow hood, what the hormones do, some of the suppliers, media recipes. I mean, it's such a Julia Child cookbook of
Hmm.
plant tissue culture. And so much of genetic engineering is bottlenecked and predicated on whether or not you can interface with the organism.
Mm.
So before you start dreaming of transgenesis, show me you can regenerate a plant from a piece of leaf,
Hmm.
right? Your target plant. Show me that you can engage with it in a manner that is conducive to those methods. Because after you get that regeneration protocol working, everything else is just like soak the leaf in some agrobacterium for a little bit and then do the regeneration as you
Yeah.
normally would,
Yeah. Yeah.
right? And so that core skill that sometimes people feel as busy work or janitorial, because they're like, no, I want to get to the genetic design. I'm just like wax on, wax off, pedaling, just like start with the basics. Like if you're learning French cooking, you start with your roux, right? You start with the sauces. And or can you make an omelet, right? That's like the equivalent of show me if you're a chef, show me you can make an omelet. And for
Mm.
me, it would be show me your tissue culture, right?
Yeah. Yeah.
And so that was such a gateway drug for me. Everything after that started clicking, and there are more suppliers now than ever before in terms of like offering these type of resources. But unfortunately, I am very geographically privileged in that in the United States, BSL-1 isn't regulated. And so a
Mm.
lot of these things can ship legally residentially. In other places, I mean, Australia is surprisingly straightforward with the regulations. You can make a PC1 laboratory in your garage. I have
Mm.
a couple of friends who have done so successfully, and they've passed their inspections and whatnot. And that's sane. But in Europe, what I'm doing would be equivalent to like a federal felony, right? And that comes with like jail time, right? Like
Mm.
putting a GFP glowing green fluorescent protein into E. coli could come with a prison sentence. Like
Mm.
that's right. That the Unfortunately the country dictates how far you can go.
Mm.
But that's why I've been embracing mutagenesis and traditional breeding into my practice as well, because I want it to be as accessible as possible.
Mm.
And I also want to showcase that not like genetic engineering is not the be all end all of interfacing with a plant.
Mm.
At the end of the day, if choice breeding and some good like loaded dice rolls can lead you to interesting outputs and then do sequencing, that's solid science. There's
Mm.
so much science you can do without the genetic engineering component. And if you must do genetic engineering, community labs are a thing, right? Like the Melbourne had a hackerspace. I think Sydney had one too. Um, and there's just opportunities also at universities. Like you could just volunteer. Like if you
Mm.
just cold email, be like, I will volunteer, I'll wash your dishes, I just want to learn how to do this, right?
Yeah.
There are ways to do that. And
Mm.
absolute worst case, a community college or just enroll in a small college and just like do a biotech program.
Mm.
Like school is the greatest accelerant to getting to being able to do genetic engineering, more
Yeah.
so than anything else. Like
Yeah.
there's no glory in doing this yourself. Like yeah, it's street cred is street cred. Like, okay, that's cool. You taught yourself, but I would rather have saved some time and learned from like master chefs than just cooking in my kitchen hoping I get it right.
Yeah. Yeah.
Because I
Yeah.
spent the last 15 years hitting my head against the wall trying to figure out how to do this. And I'm pretty sure I could have had a more abridged experience and accelerated my ambitions more quickly. But then remember when we talked about double-edged swords? I wouldn't be who I am today if I did go through the academic
Yeah. Yeah.
mill, right?
Yeah.
And so I don't know how to answer whether that's a benefit, like whether it would be something I would recommend.
It's a
I mean,
trade-off. Yeah.
it's a huge trade-off and it makes for a very interesting life. And but when people are really excited about being a pioneer in something, people forget that not all the pioneers survived that westward expansion,
Yeah.
right? Like people in the US talk about Lewis and Clark and going to the westward expansion stories. I'm like, those were two people out of so many pioneers that just like died in the forest by a bear, right?
Mm. Mm.
Yeah, so there's a cautionary tale to my life that I'm also trying to be really transparent about.
Yeah.
Because like
Yeah.
there are
Yeah.
a lot of sacrifices in the background. It's not all just like, look what I'm doing, yay.
Mm.
Yeah, it's a tremendous amount of sacrifice and a level of perseverance that I didn't know I could foster until I had to. I didn't know I loved biology until like my entire life was kind of crumbling around me, but I was still really excited to see what the colonies look like the next day, right? Yeah, I'm just like, oh, did my PCR work? Yay! Like everything's crumbling, like relatives dying, money's gone, all nut. But like I was really excited for that one scientific result. That was the moment where I'm just like, okay, I love biology. I love
Yeah.
this so much. Like I need this to be a person.
I don't know if you agree with this, but I went through an academic program and had lots of like crashes and disasters along the way too. And I think the one thing I've learned from all of that and that I continue to learn today is how to manage risk. When you take on a project, you get an intuition about whether the chances of something paying off are worth investing the time and the energy and the resources
Yeah,
that yeah, you get a weird, and you only really gain that intuition by having disasters where you inject
yeah,
a lot of energy and time into something and it all just ends up in a big pile of mess that you have to throw away and
yeah,
start again.
yeah. And I wish I could tell like the so the young players in the audience about a shortcut to this. But like there's no better teacher than failure, right? Like
Mm,
failure at this point is like my best friend. I walk with failure all the time. 99.9% of everything I do is fail.
mm,
And that's not for lack of trying, right?
mm.
And it's just we're working with this very, very fuzzy logic, kind of strangely stochastic system that we have yet to fully understand. And I don't want to use that as a cop out because you still have to try. You can also fail by just like not putting in an effort.
Well,
Um,
you're always dealing with the unknown. Like if you're doing something exciting,
yeah,
you don't really know what the outcome is going to be. So there's
yeah,
always a chance that it's going to be all for nothing, or at
yeah. A
least you'll
bunch of my
learn something that you
friends,
weren't expecting to learn.
exactly. Yeah, a bunch of my friends are just like, how do you deal with working in a completely unknown all the time? Like you're not producing, you're exploring all the time. How do you deal with that almost guarantee that it's gonna dead end somewhere?
Hmm.
And it's just I have this like 1,000 yard stare, and I'm just like, yeah, you just get used to it, right? You
No,
get you, like this,
but we got to fit in room. I've got a list of techniques that I wanted to get
please.
your perspective on, like how accessible and useful they are for amateur plant breeders. So I'll
Sure.
try and start with the simpler ones and then build up to the more complex ones. So first one I
Yeah.
want to talk about is various methods for inducing polyploidy, which is often really
Mhm.
important for breeding new crops,
Yeah. So like the go-to is like colchicine, for example,
which
from
is
the autumn
pretty
crocus.
nasty to handle.
Yeah. It's pretty nasty. Anything that is a teratogen or messes with genes will mess with yours, right? And
Hmm.
so um I'm so not a fan of chemical mutagens like at all, right?
Mm.
Even though like there's EMS, right? Ethyl methanosulfone, whatever. I'm not a
Oh,
chemist.
yeah, methylating agents are even worse. Oh,
Yeah,
yeah.
yeah. So all of these like terrible things make wonderful phenotypes. That's the biggest problem, right? And you can
Mm,
be drawn to it. But if you don't have a decent understanding of chemical hygiene, or at least like shadow somebody who does this, right?
mm,
Like don't gung-ho this because it is dangerous. And I would
yeah.
never advocate somebody to hurt themselves. And ignorance isn't innocence because in some cases, if you hurt yourself, you could then be in legal trouble because you've done this in a space that endangered others or something like that. And so
With chemical mutagens as well, you could be endangering your future children without
exactly.
realizing it too.
Yeah. And so like accessible, but should you, right? Like is that the best idea? So there are techniques that won't directly immediately do this, but have the a great potential to do so. Like for example, if you're doing tissue culture, and let's say it's 3 o'clock in the morning and you're really tired and you accidentally made 10 times too much of a hormone in your media. That happened to me a couple times. And my plants started doing really strange stuff. They were making these crazy tumors and whatnot. And whatever I regenerated from them, some of them became aneuploids, right?
Ah, mm,
Right, in the process because you're causing rapid cell division, and sometimes it desynchronizes and you get a chromosomal doubling event without the cells dividing because
mm,
like you're slamming it with cytokinins, everything's dividing all the time. It's a disaster, but it is a way safer disaster than using something that is acutely like
mm,
spindle inhibiting,
yeah.
right? Like mitosis inhibitors or meiosis inhibitors. So yeah, I would say dig into the mechanism of action and then see if there are other things, right?
Mm.
Like one another technique that I'm really interested in is microwave mutagenesis. You
Oh,
just put the
wow.
seeds in the microwave for a couple seconds and they come out all weird.
Hmm.
That's physical, that's heat stress, that's so many stressors that are hitting that piece of DNA that has a chance to either produce a lot of transposition, which is just as beneficial. These
Mm.
jumping genes can make all kinds of things happen, but also just physical damage. Like there are many ways to stress a plant without using things that could harm your future offspring.
Yeah,
Yeah,
yeah. I believe I've got some friends who are trying to use a glutamate technique, just MSG, that you can soak seeds during germination in high enough concentrations, and that can induce polyploidy, at least some of
incredible.
the
Yeah,
time. So much safer to
yeah.
handle.
Yeah. And so what I would recommend is to not care too much about the efficiency and
Mm,
rather like the trade-off, the benefits of not doing the dangerous stuff.
mm,
Like with UVC, like sure, EMS will do all kinds of mutations, and UVC will more often do pyrimidine mutations. So like TT, CT, and CC mutations, even though that's still something, and you're protecting yourself from a really, really huge, very real risk,
mm.
right? Like I even if I had access to it, I would not want to buy a puck of cobalt 60 and like some acreage in a forest and do atomic gardening. Like I just wouldn't, right?
Yeah, yeah.
Yeah. And so like I would actually encourage people to explore these things. Like who knew MSG at high concentrations would do this, right?
Mm,
What
mm,
other common household items could do something like this? What
mm,
about soaking seeds in peroxide to cause
mm.
oxidation reactions, right?
Yeah,
Um, you can do terrible things to plants without risking
your
life
own
and
health.
limb.
Yeah.
Yeah.
Just a quick side note. Are there any hazards people should consider with just tissue culture itself? I'm guessing the biggest thing would be is if nasty bacteria or fungi grow in the cultures, and then you can be exposed to high doses of them if you don't handle them carefully.
Yeah, like I wouldn't open any culture that's got contaminated, even if it's like precious tissue. Like you
Mm.
are more precious than that tissue,
Yeah,
right?
yeah.
If you're like propagating some like whatever rare means, some like rare philodendron, like a lot of people are like monsteras and philodendrons, they love propagating those, right?
Mm.
If it gets contaminated, just put it in the autoclave, just kill it, right?
Yeah.
Like don't open that. But also like the hormones themselves, right? Like there's cytokinins. There's so many systems in humans that have paralogs in plants and in insects, and like the MADS box receptors on certain promoters. They call it MADS because some of them are just like mouse and Arabidopsis and humans and snapdragons. Yeah, like all of these are there, right?
Mm,
Like we all evolved from a common ancestor, so a lot of these chemicals could also interact with you.
mm,
And so like proper PPE is very, very important,
mm.
obviously. But life is also about risk management, like you said, right? So acceptable risks. You
Yeah.
can if you wear gloves and you protect yourself and you are fully aware of what happens if you were to make contact with it, like one of the biggest opportunity hazard, like info hazards, is not reading up on the chemicals and what they do,
Yeah,
right?
yeah.
Like not having some level of chemical competence of what you're doing, not just like put in magic potion A and then things happen, right?
Mm,
Because like what if magic potion A you happen to be allergic to, right?
mm,
Because it's rare, but like it could happen, right? But more often than not, plant hormones are present in all the tissues of a plant. So
mm.
if you're eating carrot, you're eating auxins,
Yeah.
right? And cytokinins to some extent too. So we're being exposed to these things. It's not as risky, but yeah, I would worry more about like really nasty contaminants like black mold growing on your
Yeah.
thing.
It's
Um,
probably worth also pointing out that mainstream gardening has plenty of chemical hazards in it as well, that people
Absolutely.
just kind of shrug and accept. And,
Yeah.
occasionally a small child drinks something and ends up in the hospital.
Yeah, yeah.
So
Or
it's not,
even
it's
just
not
like
safe either.
and even the act of gardening, because sometimes in bad soil patches, you have molds that could be
Mm,
life-threatening,
mm,
right? That
mm.
you're engaging with. Like a lot of older folks who with compromised immune systems suddenly develop pneumonia and they don't know why. And it happens to be because you were engaging with like dry dirt that wasn't too
Yeah, yeah.
happy,
Or a
right?
compost heap that was a bit strange.
Yeah, exactly. Like don't
Mm,
put meat in your compost, right? There's
mm.
reasons.
Yeah.
But yeah, there's yeah, go ahead.
Oh, well, the next technique I wanted to talk about was embryo rescue for wide
Mm,
hybrids. So often when you cross really distantly related plants, it will start to
yeah,
form seeds, but then the hormone production doesn't quite work and
yeah,
the fruit will fall off. But there's a little tiny seed in there that is actually alive. And if you get it into tissue culture at the right time, you can grow that into a whole plant. And this was like a
yeah.
major technique, I think in the 60s and 70s. It was done all over the place to see what was possible. How accessible is that to people as home lab
So
setup?
yeah, so I love embryo rescue. It's fantastic. It gets insane if you're doing it on a Arabidopsis because it's already a tiny plant, and then you're
Mm,
rescuing tiny, tiny, tiny embryos.
mm,
But like a cheap dissection microscope off eBay, right? Enough to be able to see like 10x or 20x. In fact, there's a lot of like Alibaba or Amazon suppliers that have these microscopes that have really high magnification with a LCD screen, and everything's battery powered, and you can just put it in your laminar flow hood.
mm,
You need a microscope and fairly steady hands that you can just develop by propping it upright. Like it's a mechanically accessible technique,
hmm
right? Which I would highly recommend because there's no real risk there. Well, all you're doing is just stressing yourself out trying to pick up this really fragile piece of tissue and then
mm,
praying, right?
mm,
But yeah, I like especially with larger fruits, absolutely like peppers, eggplants, tomatoes, those type of things, please do embryo rescue. Like we're
mm,
missing out on so many interesting things because that fruit fell off the vine, right?
mm, mm,
Yeah, I would recommend that wholeheartedly. Like if you can make bridge species between really common families, like if you can make a tomato by doing grafting and just
mm.
like culturing the graft, yeah, it can get really, really funky. Somatic hybrids are incredible, and I would strongly recommend it, even though it looks scary. Like start with embryo rescue. It's a
Yeah,
gateway drug. You'll
yeah.
get to far worse things, somatic hybrids and
Well,
proto plus fusion.
that was the next thing I wanted to talk about because this technique, somatic hybridization, I'll outline for people who don't know it. In tissue culture, you take the cell of plant A and the cell of plant B, and you basically smush them together to create an allopolyploid, which you can sometimes form with hybridization. But for species where the pollination mechanisms, the sexual mechanisms are just completely incompatible, you can just crush those two cells together and then grow out a kind of Frankenstein's monster. And this has been done between grasses and broadleaf plants, like wheat and soybeans, I think have been hybridized this way into some
So crazy.
monster. There
Yeah.
seems to be almost no limits to what you can put together. But I believe this technique is a lot more tricky because you're doing it with individual cells. So
Yeah.
how accessible is that method?
So I've done somatic hybridization before, and it's a bit of a crapshoot in terms of getting them to hybridize, right?
Mm,
Getting them because they're like soap bubbles and they fuse.
mm.
But the difficulty is actually in isolating plant protoplasts, right?
Yeah, yeah,
So
yeah, yeah.
so you have leaves that you need to melt with enzymes to remove all the cell walls and debris to leave these like free floating cells. But plant cells have evolved to have this they're hydraulic creatures. So the cells push against the vacuole that wants to expand outward, causing this turgor pressure. And so when you remove the cell wall, what you get is a cell that constantly wants to expand with no container to push back on it. So
mm,
they explode.
mm,
And so what people found out is that if you densify the media by putting like a ton of a sugar that you don't normally use, like mannitol, right? And put just so much of it, like 18 moles of it, tremendous amount of sugar inside. It acts like a liquid cell wall, pushing against it with density
mm,
osmotically to keep those cells intact. Now, if you can isolate those and regenerate a whole plant from them, from those cells,
mm,
you now know that you can make protoplast with that plant, plant A.
mm,
Now, do the same process with plant B, and now you can do this magical thing where you take the two protoplasts in solution, just roughly, just kind of count them on a microscope, get a rough estimate of how much are each, and make equal combinations of them. And then you add this chemical called polyethylene glycol, PEG, which is in the United States, it's under the brand Miralax. It's a laxative, right?
mhm,
I use just the Miralax brand because it's ultra pure and food grade. People think you need to buy science stuff. You don't. Food grade is fine.
mm,
And if you add just the right amount and the stars align and a cosmic ray shoots at the right angle, you will get an even hybridization where the plant the what polyethylene glycol does is that it pushes the cells closer and closer together such that the forces are pushing at the cell membranes to fuse kind of like how soap bubbles fuse together,
mhm,
right? With enough force, two soap bubbles will just go bloop and fuse together. That's where the cell guts of the two plants coalesce. But then you have to be even more fortunate where the nuclei also fuse
mm,
because you can have different variants of hybridization, like cybrids, where you have cytosolic hybrids, where you have the guts of one cell and the nucleus of another, or dinucleated plants that eventually become the parent plants over time.
mm,
But if you're really lucky, which means you have to do many, many times this process using enzymes that can be expensive unless you approach it from a brewer's standpoint. Like cellulase is the main enzyme to digest cell walls.
mm,
You can get the super fancy Onizuka R10 stuff that's worth more than gold, or you can buy cellulase that is used in wine clearing to like clarify wine, to like digest the cellulose from there.
mm,
But either way, if you're lucky, you get a true like nuclear hybrid
mm,
where you get a mix of the two. You make a true hybrid. And from that point, you then have to be fortunate enough to be able to regenerate the emergent requirements of this plant that is not the sum of the two parts.
yeah.
Like if plant A has a very specific media requirement, plant B has almost the antithesis. How are you going to know what plant C looks like?
Yeah,
A plus
yeah, and
B.
you've only got that one cell in a mix of all the other cells to try and fish out
Yeah.
again.
And so it is though okay, it's not for the faint of heart, and I wouldn't recommend it for folks that are easily deterred by failure.
Yeah.
But the benefits are incalculable. Like
Mm,
you can make plants that no one has ever seen before. And if the chromosomal counts are right, it will be able to divide. But worst case, you have a one-off that you can make clones of
yeah.
and have this like momentary, not speciation event because it's not, it's not fecund, but there's like an opportunity for you to have just made something completely different.
Mm,
Right. And so genetic engineering kind of helps with this because a couple papers showed that there are graft hybrids that happen where you don't need to take cells and fuse them. You actually get somatic hybrids at the interface of a graft of two plants.
yeah, yeah.
And so if you can segment discs of the stem of a grafted plant and tissue culture those, there's a likelihood that you will get a plant that's a true hybrid out of
Hmm.
those.
Mm,
And so what genetic engineering does to this is let's say you make plant A resistant to one herbicide and plant B resistant to another. All you have to do is graft them together, slice the grafted area into discs and plate them on media that has both herbicides.
yeah,
So the
yeah.
only thing can survive is the ones that carry both of those traits. And that kind of drastically increases your odds
Mm,
to get your hybrid. But
yeah.
I mean, people have made tobaccos, right? They've made strange tobacco hybrids. They've made like weird apple cherry hybrids. More often than not, it's still in the same family. So I'd
Mm,
love to see really bridge species, like really wide, like the one you mentioned, the wheat and the soy,
yeah,
right? Or barley and soy.
yeah.
That's insane. That's black magic wizard work. I don't know how you did that.
Well,
It's,
it goes back to the idea of you wanting to make a petunia mutant where botanists would look at it and have no idea where it
yeah,
came from and what it's related to. Like if you're crossing monocots and dicots, that's about as wide as you could imagine.
yeah. or like succulents and bryophytes, right?
Hmm.
Just like really
Hmm.
like gymnosperms and angiosperms, right? Like really separate wildly. I mean, there is just so much. And what I'm hoping is that within your audience, and as I have conversations with folks like you who have audience like this, is try to convince the really intrepid weirdos in the audience to just push this because honestly, everybody sees this and they think it's so difficult and nobody wants to do it.
Yeah.
But
And
somebody has to do it.
and people in academia and industry can't afford to take these really crazy long shots if there isn't a paper or a
It's
product at the end of it.
Yeah,
But if you're
there's no
just
guarantee.
curious, yeah, if you're just
Yeah,
curious, it's within grasp. That's I think I'm on the same page as you as wanting to spread that message that just
yeah,
get out there and do it, people.
because there
Now,
are I would just want to say like there
yeah,
are academics who study somatic hybridization to some extent, but it's so hard to dig out that niche that they are probably the world expert on
yeah,
that concept. But
yeah,
even then, they're just focusing on just like how does that even work? Not
mm,
even like the production line of producing novel species. Like no one has that flexibility, but as an amateur, you definitely do because you're at the helm of your own ship. Yeah.
mm. Now, I want to focus on the horizontal gene transfer
Sure.
methods. So, what about agrobacterium transformation? How accessible and useful is that today
So
for
I
amateurs?
that so that is my bread and butter. I use agrobacterium all the time, right? It is the most efficient way to transfer genes into a plant. The caveat there is that it is geographically restricted in most places, right?
Mm,
It is considered a plant pathogen. A lot of folks that signed the Cartagena Agreement, the Nagoya Protocol, et cetera, those have restricted the use of it because it could spread into the soil and transform other plants, right?
mhm,
In big ass quotes, because these lab pet versions of agrobacterium kind of hate dirt, right? They're not really that hardy anymore.
mm,
The logic there is like leaning on superstition. But
mm,
if you do live in a country where you can work with agrobacterium, I highly recommend it because it's as simple as soaking leaf tissue into the bacteria, letting it sit on a media without any antibiotics or anything in the dark for like 2 days, and
mm,
then moving it to media with antibiotics and regenerating the tissue from there.
mm.
And so along with your gene of interest, you incorporate a herbicide tolerance gene such that in the media you add herbicide so all the wild non-transgenic tissue dies and whatever is transgenic tissue survives.
Yeah.
And that process is it's basically 99% tissue culture and 1% grow the bacteria that carries your gene of interest, right?
Mm,
And then like merge them together in a purposeful contamination, which is kind of a mindfuck because it forces you to do the one thing you're not supposed to do in tissue culture, which is contaminate your culture, right?
mm.
But yes, very, very accessible. Like of all the mechanisms we mentioned, it costs pennies to grow the microbe, and the being able to develop an optimized protocol for a non-model plant is significant enough even today to publish a paper on.
Yeah.
Be
Yeah.
like, I have transferred whatever green fluorescent protein into this plant that has never been engineered before. Here's how
Mm,
you do it. That's a significant contribution to the field.
yeah.
And
Yeah.
amateurs in spaces where that's legal, that's a wonderful way to get into the act of being a scientist, right?
I,
The act of generating knowledge.
I'm also guessing we're at a bit of a golden age now where you can order genetic constructs to put in agrobacterium to get into a
Yeah.
plant in a way that wasn't possible probably even a decade ago. And it may not be possible 10 years in the future if regulations change.
Yeah, yeah. And so like I'm one of my biggest fears is that one random career politician decides to see that there's like a weird gray area in biosafety level 1 and decide to regulate that in the United States.
Mm,
And then I would have to find a formal spot and pay rent for it in an industrial area to be able to do what I'm doing.
mm,
But it would essentially be the death knell for amateur genetic engineering, hook, line, and sinker, because then we would be equivalent to Europe in
yeah.
that case.
Yeah.
So that's always really terrifying. But not thinking about the scary stuff. Currently, right now, retail price for DNA is 7 cents per base pair, right?
Mm,
And so there are companies like Twist Biosciences and GenScript, a couple other ones that just do synthesis fairly cheap, and they will just ship to wherever you are. And so you can design on a laptop the genes so you don't have to like systematically combine them from borrowing from different places. You can just author it, send it to ship, and it'll come in 3 to 5 days. Um, and that's been like I hate to use the AI word game changer because that's always a tell, but it has been a truly a game changer because there's just so much. it opens the door to everything at that point because you can
mm,
have genes from octopuses and cuttlefish and sequoias and whatever the case may be. But that's when environmental stewardship and responsibility comes in. It's like, yes, you have access to this, and no, they're not going to let you print Ebola or do something dumb, right?
mm,
If the gene fragment you're trying to synthesize gets flagged, you'll just get your account canceled.
mm,
Right. If it looks like something, if it even smells remotely like something that's on a list of bad things, yeah, you're going to get your account canceled.
mm,
But within that realm, you can have a what's it, an ethical sandbox to explore as long as you have your own personal practice, regardless of the current administration in the US and what's happening, or wherever you are, assuming that you're following the law and it is legal in your country to do so, or more importantly, it's not legal. Because very rarely will you get your legislature that says you absolutely can do this in your home.
mm,
Like currently in the US, there's nowhere that says you can't
mm,
because it was never considered, right? So gray area. But um, within those confines, if you can, you always have to ask yourself, should you,
yeah,
right?
yeah,
Like, oh, I want to make this grass that grows super, super fast. It's like, okay, you're essentially building a weed at that point
mm,
that can spread very rapidly. Like, yes, it'll sequester CO2, but it could also swamp out an ecosystem. And how can you prevent release? Like biocontainment is a thing.
mm,
That's actually one of the reasons why I don't want to work with Arabidopsis. It's because the model organism of plants, because the seeds are like dust and they
mm.
electrostatically cling to your shirt, to your shoes. Like almost every plant department that I've ever visited had a wild Arabidopsis growing on the outside. And I'm just like, wonder how that happened, right? Like it's hard to biocontain dust,
Yeah,
right? So I do one of my on my dream canvas organism checklist was seeds that are not dust. Like
yeah,
that is a line item on it, right?
yeah,
So like I'm not always thinking about it because now it's on autopilot, but for a long time I didn't want to ruin a good time
mm,
by like stepping out of the bounds from what I thought was sensible.
yeah,
Because there is like ambition and then there is self-discipline,
yeah,
right? Like a lot there's this weird vibe because of this like San Francisco AI cult thing where you can just do things, right?
hmm,
People say that a lot now. And I feel like when it comes to self-replicating nanomachines that could escape your laboratory, I think it'd be nice to not just do things, to
mm,
have a conversation with people. That's why I really strongly advocate for DIT bio instead of DIY. Instead of do it yourself, do it together.
yeah,
Have
yeah,
like an extra person, right,
mm,
having a conversation or work in a community lab where you can have this type of discussion
mm,
because it's really easy to fall into your own ambitions. And some people's personalities are kind of addicted to that concept of power, right?
mm, mm,
Because it's that Spider-Man quote, right? With great power comes great responsibility. But it is true because outside of the fairy tales that Europe wants to talk about how GMOs are toxic inherently somehow, even
mm,
though like the tomatoes they eat are New World tomatoes that came from South America. Where is that in Germany? I don't know. Um, but like outside of the weird superstitions, there is, a viable risk of like, could this agriculturally harm us?
yeah,
Could this do anything bad? I mean, it's so hard to do something bad because like it's so hard to do something that works in
at
biology
all,
that
yeah,
like
yeah,
yeah that you need this like evil Mozart genius to like come up with this like really complex mechanism of doing like great harm with this. So more often than not, everything's going to fizzle out. But what I've noticed is that with unprompted, so many of the people within like the amateur biology community, like overwhelming majority just want to make interesting things.
mm,
They just want to create, and many more often than not, they want to create together. Very few are destructive, and those people tend to be self-ostracized. We tend to ostracize those folks
mm.
fairly quickly for whatever's left of the DIY bio community.
I was
Um,
thinking of the other Spider-Man quote. I'm also something of a scientist. The guy who's
yeah,
actually like
yeah,
the evil Green Goblin guy on the
yeah,
side. Now we
yeah,
have to talk about your gene gun transgenesis because you've
sure,
actually done this, and
yeah,
it's such a superhero villain kind of concept that you've got this like
the dream
gun
gun.
that shoots genes.
Yeah. Oh man. Yeah, so that started with learning that biolistics, which is the fancy term of using genetic delivery through a kinetic payload, right?
Hmm.
That mechanism of transformation around the time I started genetic engineering was one of the few loopholes in regulations that
Mm,
if you make it with a biolistic rather than with agrobacterium, it wasn't regulated in the same way, right? Because we cared in the US, we cared more about how you made the GMO more so than what the GMO actually was.
mm,
because they were saying anything made with agrobacterium could have latent agrobacterium on it, therefore be a vector for that pathogen,
Hmm.
right? And through that strange logic that was mostly crafted by Monsanto hand in hand with the regulators to give them a little slap on the wrist, they basically made this such that it's almost impossible to deregulate with agrobacterium, right?
Mm.
And so lo and behold came a new model way to transform biolistics, right?
Yeah.
So it started with this thing called the PIG, the particle inflow gun, right? And and basically what a gene gun is, it's a genetic shotgun. You have very, very dense molecules, particles, like nanoparticles, about 800 nanometers across.
Mm.
Uh, Typically gold because it's inert, but I have deep ethical issues with shooting gold and throwing it in the toilet.
Mm.
So I use tungsten, right?
Hmm.
Just a little bit to the left on the periodic table, and it allows a similar level of density to be had, right? So like gold, lead, and tungsten are the most dense metals that we have that are not radioactive.
Mm.
Lead is highly toxic. Gold is extremely expensive. Tungsten is reactive and cheaper, and there's some caveats, but it's still serviceable.
Mm.
And so what you do is you take DNA and you prepare your tungsten by like washing it like crazy with alcohol to like get it as dry and clean as possible. And then you add salmon sperm, which is strange. So there's this chemical called spermidine, which is like the main liquid component in salmon sperm that
Mm.
happens to be very genetically sticky, right? So if you can imagine, I mean, I don't want you to imagine it, but humor me. There's like a female salmon that lays eggs on the ground in a stream, and then the male kind of crop dusts its semen on top of it, right? And so that thing has to be dense and it has to adhere to any other biological surface as quickly as possible, in aqueous environment. So
Mm.
salmon have evolved an ideal thing to tie DNA to other stuff, right? And so we use the spermidine to glue particles of fragments of DNA to the tungsten. You let it dry on this little piece of plastic, and then you load it into this contraption that, by way of high pressure helium gas, accelerates that piece of plastic onto a mesh at extreme velocity. And so once that plastic
Ah.
hits the mesh like that,
Yeah.
the particles continue downward onto the open-faced petri dish where your tissue is.
Mm.
So you're like physically blasting it with nanoparticles
Mm.
taped with your DNA. It's extremely crude. The mechanism of actual integration is hotly debated, right? There are people who are like theory crafting around what it could be, and one of them was like, "Oh, it physically occupies space within the chloroplast, and that's how we get chloroplast transforms." And I'm like, chloroplast are 2 micron by 1 micron, and you're putting almost 1 micron worth of a cannonball inside there.
Mm.
Like that just
Mm.
doesn't make sense. So one of the prevailing theories is like a shock the physical shock of it causes ancient DNA uptake machinery, stress machinery, to make the chloroplast or the nucleus take in DNA, right? It's hand wavy. I'm not super up to date with like the latest in that, but there's also not a big impetus because it just works,
Hmm.
right? Just like pew pew, and then I mean the recovery rate is awful,
Mm.
right? Because in ground zero, at the very center bore of the shot, you get total obliteration. And then on the periphery, you get no shot. But somewhere in between, in that diameter of tissue that you arranged in the center of your petri dish, you get just the right amount of penetrance to get just the right angle with just the right amount of DNA in it to get integration events,
Mm.
right? Through essentially green magic. Like I just, I can't describe the biophysics there. I have no idea. I'm not a physicist, but anyway, so the recovery rate is like 0.001%, right? So like less, it's worse than 1 in 10,000, 1 in 1,000 if you're like working in tobacco and you've dialed in the protocol perfectly.
Mm.
But in that comes this like really miraculous thing where you can genetically engineer the chloroplast of a plant, right? And if you can engineer the chloroplast, you can produce proteins on levels that we've never seen before
Mm.
because their prokaryotic systems are little bacteria that live inside of the cell, and there's so many more than a single nucleus. So if you transform a chloroplast and you have some antibiotic resistance to kind of kill the not transformed chloroplast, you eventually accumulate a lot of those transgenic chloroplasts. So you have many copies of that gene being expressed.
Mm.
And so if you want to produce, let's say, vaccines in plants, or if you want to produce like high value catalysts or proteins in plants, chloroplast engineering is the way to go.
Mm.
But again, it's this black magic of passing through the cell wall, the cell membrane, the two membrane layers of the chloroplast, and then it gets into the chloroplast genome through magic.
Mm.
And so coming back towards me, I went online and I looked up how much a gene gun costs, and the Biorad PDS 1000 cost $25,000, right? It was a prototype that was priced in order to dissuade people from buying it, but people still bought it.
Yeah.
And there hasn't really been much advances ever since with that. But as soon as that patent expired, this Chinese company made like a really fantastic knockoff of that one that had a lot of quality of life upgrades, and it looks like an espresso machine. And they were selling it for like 4,000. And I tried making my own gene gun, right, out of some papers from like the 80s. And one of them involved like a physical blank of a pistol,
Mm.
like a nail gun blank. So it was an actual gun, and that was crazy. I'm never gonna do that again. I tried it once; it was insane.
Mm.
There are ones where you just kind of pump a pipe full of high pressure air, and that just kind of accelerates it really
Mm.
fast. They worked, but it was just so messy and so not fun, but more importantly, it was dangerous.
Yeah.
So the pipes I was using wasn't rated for that pressure. In fact, nothing that I could find could be rated for that pressure.
Mm.
So even the instructions on the manual said like it's a pipe bomb, be
Yeah.
careful.
Yeah.
Um, so that kind of dissuaded me because I tried it a couple times, it worked, but the whole time I was looking at it, I'm like, if that explodes, it'll shrapnel everywhere,
Yeah.
right?
Yeah.
Like that could kill me.
Mm.
And so as much as I'm a huge DIY a proponent, I was very much not interested in making a pipe bomb, right?
Mm.
Um, so I found online through like random musings one day of just going like Gene Gun China, right? I saw that they were producing this knockoff for 4 grand, and so I saved up for the whole year, like working odd jobs, doing whatever I could to save up. And I was like one of the first people on the Eastern Seaboard to buy this Gene Gun because it
Mm.
was China at the time where their label was like shoddy or weird or like knockoffs that have some caveats that you're not familiar with, right? And so when I spoke with academics, they said something to the effect of, I don't trust it. It's too cheap to be true, right?
Uh.
And so I said, well, the alternative is a luxury car. So no, I'm gonna buy this thing. And yeah, and I kind of like taught myself how to work with Alibaba and all the brokerage and whatnot, and communicating with these folks to like give me a discount. Because at
Mm.
first they were highballing me for 9,000, and then I told them
Uh.
like, no one will buy it. I'm an American. I can sing your praises if you give me a good discount and say
Mm.
like, give an example of one person doing it, and then more academics could come in, right, as an example.
Mm.
And if eventually they gave it to me for 4 grand, and yeah, and I have it to this day. I use it often. A lot of my client work is bialistic. Um, Whenever a plant or fungi, in some cases, are so recalcitrant to everything, including agrobacterium, the
Mm.
only alternative is to shoot it with a genetic shotgun. Like nothing else works.
Mhm.
But yeah, it is a very weird esoteric piece of equipment. The last thing I would call it is accessible,
Yeah.
primarily because the company that was selling it went out of business, and now this holdings company is selling these devices for like 13, 14,000, right? So it became prohibitively expensive. And the alternatives right now, like if there's a mechanical engineer in the audience who wants to make a safe, high pressure espresso machine that shoots it into plants, I'm all ears.
Mm.
Yeah. If safety is at the forefront and you can achieve those velocities without compromising without making a pipe bomb,
Mm.
that could really, really accelerate this. And you could sell it as well. Just don't do 13,000, right? Please. Uh, then you're just as bad as everyone else, right?
Yeah.
But yeah, if done safely, biolistics is one of the strangest and most powerful tools for genetic engineering.
Mm. Mm. Now we have to talk about CRISPR because everyone has heard the name and like heard all of the
Sure.
hype and excitement around it. How does that work in the hands of an amateur plant biotechnologist?
Yeah. So there have been some really fantastic advances with transgene-free editing, right? So one of the hottest topics in academia right now is can you modify a plant without making it permanent transgenic, right? Um, Typically, what you would do is you would have the Cas9 protein in an expression cassette, and right next to it, an expression cassette that produces a ton of the guide RNA, right? And you transfer that via agrobacterium permanently into the plant, and the plant's just going to keep on making Cas9 and the guides all the time,
Mm.
or ideally inducibly, like add some alcohol and starts producing it. And then what would happen is that the Cas9 would be trained by the guide RNA, would take up the guide RNA, and then skim and look for that gene, that section of interest and snip it. And the non-homologous end joining aspect of DNA repair in plants makes it such that when it glues back together, it kind of makes a a crappy job of it, and it breaks the sequence. But by doing so, you constantly are making a Cas9 expressing plant. Like you need a Cas9 line and then transiently express the guides. And that's very messy. And so
Mm.
what people are doing now is trying to utilize defanged plant viruses as a vehicle for momentarily delivering the Cas9 and guide RNA expression machinery to do the edit, which will be permanent, but then the machinery drifts away because it's not, it's an ephemeral piece. It's a
Mm.
momentarily there mechanism. But then I just said the V word, virus, right?
Mm-hmm.
So from an amateur perspective, there are so many things, especially in different countries, where if you say that you're just like actively working, even with disarmed viruses, especially post-COVID, yeah, it's a nightmare.
Mm.
So we go back to the dark green arts of protoplasts, right? So what you can do is in E. coli express Cas9, and in E. coli produce a ton of the RNA the guide RNA, purify it out, combine them in a tube, and then add them to protoplasts with polyethylene glycol,
Mm-hmm.
where it'll just kind of shove the ribonuclear complex into the plant. It'll do the edit, and then you have to regenerate from a single cell.
Mm.
So you've now added two layers of complexity. Do you can you CRISPR well? Like how's your CRISPR foo? Are your guides targetable enough? Are they specific enough? Will off targets be a thing? Like you need to know the genome of the organism.
Mm.
But then you also need to be kind of a protoplast master. But when you harmonize those two together, the output is a non-transgenic, permanently edited plant,
Mm-hmm.
right? Which is miraculous because you can bring it to market immediately,
Yeah.
right? So if you know the gene you're targeting and it's worth your effort, you can knock down that gene and make that go away, and it looks like nothing happened, right? There's
Mm-hmm.
no the surgical tools no longer remain inside the patient, you know.
Mm-hmm.
And though that is like level 9 wizardry, and it's accessible on paper, right? Because you're just stapling two methods together. But you'd have to cut your teeth on both of these things simultaneously, right? Like you have to make an expression cassette in E. coli, you have to know how to purify RNA, you have to be very good at protoplast isolation, but more
Mm.
importantly, very, very good at protoplast regeneration. Because the amount of protoplasts that are going to be taken up by the Cas9 and the amount of those that have successfully edited that segment of DNA that you're interested in
Mm.
drops by orders of magnitude.
Yeah.
So
Yeah.
you have to be able to regenerate all of the cells that you touch. You need like a green Midas touch to be able
Yeah.
to do
Yeah.
this.
Yeah.
Yeah, I'm not trying to like scare
It's
people,
a
but
high level technique.
it's a very high level technique, but the output is miraculous,
Mm.
right?
Mm.
And so because the output is as if you did it with traditional breeding.
Yeah.
Because right now, the letter of the law in many countries is if you could have done this with traditional breeding, and this is just an accelerant in terms of time, then
Mm.
technically it's not regulated,
Mm-hmm.
right? Like in the European Union right now, 20 edits or less is totally kosher. They're
Mm.
fine with that, right?
Mm.
Anything more than this arbitrary number, what is 20? I don't, when you hybridize plants, you get bajillions of rearrangements, massive chromosomal, yeah. Crossing a plant with pollen does infinitely more changes, but for some reason, high precision edits are verboten in
Mm.
Germany and
Mm.
all the other places. So, while that is frustrating, the edits themselves are also limiting because CRISPR is really good as a scissor. It can cut well, it can disrupt. But what
Mm.
if I want to add something,
Mm-hmm.
right? Like doing a knock-in with CRISPR is even more difficult because
Yeah.
on top of having that complex, you also have little fragments of the gene you want to knock in conveniently near the nucleus, right? So there's just there's compounding levels of difficulty to get that to work.
Mm.
And then once you put the gene in there, sadly, now it's just a classical transgenic organism because
Mm.
where'd that gene come from,
Yeah.
right? Like it had to come from somewhere. Yeah. So CRISPR is accessible now more than ever with mechanisms like defanged viruses or the protoplast method. Um, but again, the techniques there are pretty massive.
Mm.
Yeah.
Mm.
What I'm most interested in right now, just to add like one technique, topical RNAi,
Mm.
right? So there's a bunch of companies that are using them as biopesticides, where you make this little sandwich of double-stranded RNA by just mirroring a piece of a gene forward and reverse, and then they just
Mm-hmm.
fold together. And then just spray that on the plant, and it will uptake that DNA, I mean that RNA, and will do the silencing. You'll train it to have its own internal CRISPR called dicer that will silence it. And it's like Nobel Prize winning stuff, and the patents are off, right? And so that's a bit more, it's transient, If you do it through agrobacterium, it becomes stable. But
Mm.
either way, it's there's no patents and it's infinitely easier than all the moving parts I just mentioned.
Yeah, yeah. Well, I mean, I wanted to briefly talk about the possibility of doing horizontal gene transfer without tissue culture. Is that possible? And are there t methods there that have potential to be developed more so that you don't have
Yeah,
to set up a whole lab? If you're just growing the plants and have some extra magical ingredients, you can just fast forward to
yeah. So
HGT.
there, so there is there's another hot topic called the cut, dip, bud method, where essentially using plants that produce rhizomes that can produce shoots from rhizomes, you essentially take a cutting and you dip it in a special type of a cousin to agrobacterium called Rhizobacterium rhizogens, right? And this one, instead of producing giant tumors on trees, it produces lots of roots,
Hmm.
right? Transgenic roots. So it'll transfer your gene of interest and also make roots as if you dipped it in rooting hormone, right? And if you add like a visual marker, you're going to have white roots and then like red roots or green, glowing green
Mm,
or whatever. Then from those roots, depending on the species, you can make shoots come out of those roots, like in poplar or in strawberries, for example. Like there's a couple species, and now they're increasing the repertoire of this cut dip method.
mm,
If you can regenerate from roots, you no longer need a lab.
mm,
Right. You just need to grow the bacteria. It could even be kind of like a crappy, dirty culture in open air because the bacteria grows fast enough. And just like dip the thing in there and then wait. And if any of the roots come out red, like nurture those roots, see if you can get shoots out of those. And then that's a transgenic plant.
mm.
Um, and so that's also accessible. The key being you need to have access to that strain,
Yeah.
right? And I would have put it on my store, but it's like it's still regulated as a plant pathogen, so it can't cross state lines, let alone international transport.
Mm,
Agrobacterium tumefaciens, the one that is used to transfer DNA, that's been deregulated in the United States
mm,
for interstate transport, so I can ship it around no problem. But the rhizogens I can't without a APHIS permit and a bunch of paperwork. And so, unfortunately, like yes, on paper, it's very accessible, but you need to find that strain.
mm,
Now, that strain came from nature. So there's a very high likelihood that you could, if given enough time, isolate your own agrobacterium or your own rhizogens from there using like hairy root culture, where you just like soak a bunch of the microbes you isolated from soil with a bunch of carrots and see if roots come out of the carrot pieces.
ah,
And if it
yes.
does, score, you got it, right? And that you can follow like the paper that that strain came from will help you figure out how to find similar strains in your local environment.
Hmm.
That adds a whole side quest campaign of adventure to find these things. But you can, if you are diligent enough, you can make a YouTube series of going, here's how I found agrobacterium in the wild and I
Of
domesticated it.
residues. Yeah. I mean,
Yeah,
ironically, sourcing new wild strains and putting them in culture is more likely to cause the problem that they're worried about pathogenic versions versus the domesticated lab versions that don't want to go back
yeah,
out into the wild. But anyway, that's
right.
another point.
Yeah.
Another technique I've kind of picked up on is during pollination, there's some porousness to absorbing foreign DNA and
Yeah.
incorporating it into the seeds as they form. Have you seen anyone
Yeah.
working on those techniques?
Yeah. So I've seen some like one-off papers of like really, really wild reports where so let me start with a bit of a horror story. Horror in the sense of like a waste of time.
Mm,
So there was a paper out that called for pollen magnetofection, which means that they used magnets and magnetic particles that are bound to DNA to
mm,
pull DNA into pollen by putting a magnet right below the pollen. So you layer it on the bottom of a solution, you put a really strong magnet below it, and then you put DNA bound to magnetic particles on top, and it'll just kind of pull into those pores you mentioned.
mm.
And then you take that pollen and you cross it into a plant. That later was found to be fabricated. A bunch of the electron
Uh,
microscopy was copied and pasted. Elizabeth
mhm,
Bick did a thing on it. Yeah, PubPeer just tore it to pieces, right? That kind of like DNA, I mean the Science Sleuth organization. Yeah, so that kind of wasted a lot of people's time. But then at the same time, I keep seeing publications of like maybe there's glimmers of hope
mm,
that if you have just a strong enough magnet, or if you have the right buffer that keeps the pollen viable, but also allows the DNA to ingress at high concentrations, you could do that.
mm.
The thing is, it could be possible, like every now and then, but for it to be a method, it has to be reproducible,
Yeah.
reliable enough, right? And so like freaky one-offs, I'm a big fan of, right? But like I wouldn't confidently say that any of the pollen transformation technologies I've seen today could be labeled as like a viable protocol. Because
Mm.
otherwise, everyone would be doing it. Because
Yeah,
nobody
yeah.
wants to do tissue culture for 4 months when you can just lob some DNA onto pollen, right?
My suspicion, this is just my instinct if I had like a whole other life to work on this, is that the correct conditions on the actual stigma
Mhm.
could be the place that would be the most convenient to do that. Some kind of addition of foreign DNA and some solution to change
Yeah,
the conditions while the pollen is germinating. There's that little window of susceptibility to incorporate the DNA in the pollen
yeah.
nucleus, and then it finds its way down into the seeds. But it would be starting from almost nothing. I think there's some reports of doing something similar in the 70s with maize, where they were
Mhm.
getting what looked like a transformation, but yeah, that's a very old paper.
Yeah. What I'm interested now more than ever is actually like expanding the repertoire of viable species for floral dip.
Hmm.
Right? So floral dip's an agrobacterium method that's really established in with Arabidopsis, where essentially you wait for the flowers to bloom, and at a specific stage of bloom density, you just dip it into a vat of agrobacterium,
Mm,
and with some really, really powerful fluorinated surfactant that kind of just breaks the surface tension of water to the point where the bacteria can like slink in to the undeveloped flower bud and into the
mm,
ovules
mm,
and transform the ovule so that when it self-pollinates, you now have a Mendelianly recessive transgenic population.
mm,
Um, that works for radish, some mustard, and Arabidopsis, but they're all
hmm.
kind of mustardy,
Yeah.
right? They're all in that like turnipy area. Um, I would love to see this on petunia in nightshades. In like I would love to see more explorations of floral dip because
Yeah.
that would unlock so much because pollen is finicky, but ovules are very robust,
Mm,
right? I mean, at one point, I even thought of like ultra low concentration agrobacterium physically injected with the thinnest needle I can into the ovaries, into the oviduct of it,
I
and just
I reckon
transform them.
cucurbits would be the place to do that because you've got such huge chunky flowers that the
Yeah,
just the physical manipulation would be
exactly.
feasible.
Yeah.
Yeah.
Yeah, and maybe like either immunosuppressants or like vitamin C to prevent oxidation, right? Because
Yeah.
you are flooding one of the most privileged and pristine tissues in the plant, and who's adapted all kinds of things to protect it from death, including the easiest one for the plant, which is like induced necrosis.
Yeah.
Like, oh, that flower's gone. Like, I'm
Just
just gonna
drop it
shut it
off and
off,
try
right?
again. Yeah.
Yeah. And so though that sounds insane, it still is viable because that's how floral dip works, right?
Mm,
It just targets at a particular developmental window.
mm,
The monastic pursuit here is that you need to find that developmental window, which varies greatly. Like what I've learned in studying flowering locust tea, the main protein that induces flowering, they managed to find that there's a very specific developmental window that was hitherto unknown up until that publication in Arabidopsis, because they could visualize the formation of the flower early. But that window is different from species to species. But if we can identify that phase, maybe like in Petunia when it's like just about to unfurl, or at some point, we
mm,
could do something similar.
mm,
But unfortunately, the fortunate for Arabidopsis is that the flower matures at a pace in which there's a moment where the ovules aren't sealed, where the ovules are open, right?
mm,
And so if you can find that moment and flood it with agrobacterium, theoretically you can do it with a needle. You don't have to
mm,
just like crudely dip the whole plant and be like, I don't know, 10,000 seeds, one of them's going to be fine. Yeah, because that's like the name of the game with floral dip now is numbers, right?
mm,
So if you have one construct, you're going to dip, you know, maybe tens of plants carrying hundreds or thousands of flowers and hoping that one of those are transgenic, right?
mm, mm,
Because the percentage is like zero point 0.1 if you're great,
mm,
right?
mm,
And even that, like the conditions to get it perfect is a nightmare.
mm.
So there are techniques that we can expand by first starting with the things that work and then go outward. Because I mean, no one's floral dipped a cactus, I'm sure, because like they bloom like once every never, right? Um, but what if there's a more viable way to do transgenesis in that, right?
Mm,
Because as the CRISPR patents are expiring, more people will be really intrepid and ambitious to do gene edits, right?
yeah, yeah.
And I feel like the landscape is changing with the that superstition about GM equals bad,
Mm,
especially in ornamentals where you like don't eat my roses, right?
mhm,
Like
mhm,
don't chew on my daisies and you're fine. Yeah, there's a lot of room for this like timeless. Like I starting to feel that as we progress with technology, more older technologies become timeless
Mm.
and become deeply synergistic with modern things. So like I don't like it when people say, "Oh, why are you doing zinc fingers or why are you doing talons? That's old technology." I'm just like, it's patent free,
Yeah,
right?
yeah,
Like it and it works. It was annoying back when synthesis was really expensive to make talons, but now I can print a talon array for like $40,
yeah,
right? Why aren't we doing more talon stuff? Why aren't we
yeah.
doing more zinc fingers, more RNAi stuff?
It's that
And
it's that pursuit of trendiness, like
yeah,
the latest fashion in academia that is
yeah.
it's so
And
restricting.
yeah, and for me, I would ask like, do you want the cool flowers or do you want to be trendy, right?
Yeah.
Like, and I mean, for research purposes, I understand CRISPR because if I had total freedom to operate, it's a wonderful, simple tool
Mm,
where the same coding protein with a tiny, tiny sequence of DNA that then turns into RNA can target multiple regions
mm,
at once. You can like carpet bomb the genome with CRISPR deletions. The stuff that you can do, like I'm jealous about the mammalian culture people because they can hold a cell with a little sucker and it's fine, and then you can inject the CRISPR complex into it and it just does it, right? And people are like, "Oh, human engineering is so hard." I'm like, "Ah, please, please, spare me." Compared to the plant wizardry that I mentioned, the human stuff is a cakewalk, right? And so I'm so jealous of those folks who just can like knock down genes at will, and I'm
mm.
staring at my highly polyploid heterozygous hellscape, right? And going like, I need to knock down seven of the same gene in order for this to work,
Yeah.
right?
Well,
Yeah,
just a little note. A really good case study of what can be stumbled into by amateurs. I don't know if you heard about the cacti collecting people figured out if you put concrete dust on the stigma at pollination, it seems to break down all of the hybridization barriers and self-pollination barriers. And it's such a simple
what?
technique. And how somebody just stumbled into doing that. I mean, it's probably a pH change or the minerals in
Yeah,
the
yeah,
dust.
just like killing the S locus proteins, the self
Yeah. Yeah,
RNases. Yeah,
and it's
wow,
such a great example of what is possible to just stumble into with what could be. I mean, you could do that based on principle of understanding the biochemistry of what's going on, or you
sure,
can just randomly stumble into it and have a whole
yeah,
new set of
because
techniques open up.
I'm sure that person was like drilling in drapery onto their drywall next to their thing, and then had to also pollinate the cactus or something. Because
Yeah.
like concrete dust is so specific, right?
Or,
Like
or
it's
they
not
had
like plaster.
or they had some kind of vivid dream, like the guy with the benzene structure that he
Yeah, yeah,
figured
yeah.
out that way. I have another question too. Have you
Sure.
seen much potential in manipulating the microbiome of plants? Because there's all these endophytes, fungi, and bacteria that live inside and on plants that we've only just realized are there with more advanced DNA screening methods. Has
Yeah.
any a lot of them are difficult to culture, so it's some trickiness there. But
Yeah,
yeah, is there potential for manipulating the plant microbiome rather than the nuclear
definitely.
genome?
Yeah, definitely. So a lot of really big ag companies are for a while now. I don't know if they've divested from it, but a lot of them have been focusing on the microbiome as the nitrogen fixation alternative, for example,
Hmm.
right? Like if we want drought tolerance, what if we just recruit more drought tolerant microbes that can help like make a hydration layer around the roots or something in the event of a drought? Can
Mm,
we make because we can engineer microbes way, way better than we can with plants. And the deregulation landscape for microbes is drastically different, like
mm,
enormously different, because you're also not eating those microbes.
mm.
And so, like the mucilage in corn, right? So corn aerial roots, some species, especially the ones in Mexico, secrete this like goo that lacks a formal nitrogen source, so that the only thing that can benefit from the other nutrients there is something that naturally fixes nitrogen.
Mm.
So you're making like a nitrogenase trap that like harbors in things that can breathe in nitrogen from the air, and in exchange for like an oxygen-free environment and free starch that it can eat. Um, Modifying those microbes to be like more hardy to like disease, more hardy to drought, to cold. Definitely a really interesting route. The catch there is that like microbiomes are so complicated to make like to control a microbe that you've engineered that more often than not is a lab strain,
Yeah.
because the wild strains are just so feral and they just don't care about your antibiotics, or they don't care about your genetic engineering methods. Like you take this lab strain, you put it in the dirt, and it's just going to get wrecked by phages or outcompeted by the native stuff. And so there's this like microbiome ecology happening like system level where the rhizosphere is the main point of focus, but it in and of itself is its own cosmos
Mm,
of like archaea and fungi and bacteria that the prevailing notion is that if you dose enough of it, you'll get the effect you want because you're just swamping that area with this microbe.
mm,
But then at the same time, the plants or the surrounding flora could react in a different, interesting way as
mm,
a result of this mega dose of this microbe. Um, so there's a lot of variation. It's such an interesting field. The it's just so many layers of complexity. It feels like a Rube Goldberg apparatus,
yeah,
right? Where you have all
yeah,
these like contraptions just to turn on a light, right?
yeah.
But for pragmatic purposes, it makes sense because if you have drought, however you manifest drought tolerance on the microbial level, if that can just like cake the roots in some protective goo, right, before a drought comes, and the final result is your crops survive, I'm
Mm,
all for it. I don't care how
mm,
complicated it is. Yeah.
mm, mm,
It's just from an amateur's perspective, you would have to do a lot of sequencing. You would have to like wrangle synthetic communities in a in a wild area. And then also you have to consider the deregulation aspect because to truly prove it, you would have to have a field trial where your microbe won't escape. And I don't know, once it goes outside, it's outside,
mm,
right? Like
mm.
like the dirt is a continuum to the aquifer. It will go to the sides. Like I can't more often than not, it'll probably just die. But
Yeah.
once it's outside, we are beyond biocontainment. And so I had the privilege of actually being at a anniversary of the Asilomar conference in Asilomar in California, where they first dictated what biosafety levels are and how to contain biology as the burgeoning field of genetic engineering was forming. And one of the sections that I was a part of was actually on beyond biocontainment. Can we make a new biosafety level system that allows for intentful release of genetically engineered organisms, knowing fair well that once it's out, it is out,
Mm,
right?
mm.
Because we can't operate on the assumption that we're engineering all of these microbes, but they're going to somehow only stay in your field. Like that would be crazy, right? That's just such an impossibility. And so I think walking hand in hand with folks that are deeply thinking about this could allow us to do rapid field tests to
Mm,
finally prove our hypothesis, where the risk, the cost benefit analysis is sane and
mm,
all the stakeholders are notified, right?
mm.
Like if the town is notified that they're going to try this new microbe and the town council is like, sure, let's do it. It'll protect our corn. Great. But there's a lot of there's a lot of complexity, and with that complexity requires multiple stakeholders, especially when it comes to the fact that you are purposefully releasing this microbe
Yeah,
that has been
yeah.
engineered, you know? Yeah, and that changes the entire paradigm of what we thought biosecurity was. Because the
Mm,
whole idea is how can we keep the bug in? And now we're going, okay, so we want to put the bug out, but we want to do it in a safe way. How do we do that?
mm.
It's a fascinating question. Everybody at the conference had really fantastic feedbacks and inputs, and it led to an entreaty list of recommendations for beyond biocontainment that was made public. But even then, like there's still huge open questions, right? Like how do we make a pyramid of ultra high risk to negligible?
Yeah,
Because that means that amateurs can work in that negligible space, which is an infinite sandbox of possibilities. Like define my sandbox and I will be satisfied, but don't tell me that you don't know where the edges are, but I can't do it because you don't know the edges, right?
yeah,
Like tell
yeah.
me where it's safe and I'll play there, you know?
It
Yeah, and
makes me
so
wonder, like you mentioned that facility with the cobalt making new chrysanthemum strains.
yeah,
They're probably mutating the microbiome as well as the nuclear
absolutely.
genome, but nobody's
Yeah.
like we only realized that there's all these symbionts living in plants like yesterday, and now we're
Right.
like reassessing like everything we've been doing for the last 100 years
Yeah.
in that
Like
light.
wearing my microbiology hat, I would love to take soil samples of those runs because I'm sure like really close to the cobalt 60, everything is sterile. But in
Mm,
the outskirts of that is just an ocean of change,
mm.
right? And it'll also alter the viruses that keep steady state the microbes in that environment as well, right? And so like this I can see this like super nerdy predator prey analysis of cobalt 60 impacting a forest,
Yeah,
right?
yeah.
Of just like all of the things that change. I mean, infinite
Mm,
questions. But
mm,
the
mm,
but there's also because there's infinite questions, there's also infinite potential to find novelty within that space.
mm,
Like I sincerely doubt that the company that's doing the chrysanthemums even cares about the dirt immediately below that puck, right?
mm.
Because radiation happens in a big old circle. So we're getting things deep down as well. Yeah, we're missing out. And I feel like the only way to really harness all of the potential that formal academics could not, in good faith, ever explore to its totality, because there's only so many scientists on the planet,
Mm.
is to kind of champion for more people doing this at any level, right? Like there's literally an infinite continuum of questions and species that you can isolate and whatnot, that there's no such thing as biology being solved right
Mm.
now,
Mm.
right? And not for a really long time. Even if AI like swallows the sun, there's just like evolution is happening all the time,
Yeah.
right? There's
Yeah.
constant speciation events, and there's just so much difference. Like again, the stuff on your shoes right now could be incredibly interesting things, and we will never know. And
That's
so
why I
my
love seeing the tech bros like saying, "Oh, we've got the genomes. We'll just feed them into an LLM, and then biology is
yeah,
solved." And it's like you have
yeah.
no idea. You have no
So
idea.
there was a science a Nature paper that came out I think in 2011 or 2016 that said there are 2.87 million species of eukaryotes on the planet, and over 86% have yet to be described.
Hmm.
Right? Like in the environmental DNA, they're finding these sequences like protists and like all kinds of weird eukaryotes. But that's eukaryotes. The
Mm.
prokaryotes are almost infinite. Because like, what is a species to a prokaryote? It's
Yeah.
just like strange melange of things that can horizontally transfer with anything, right?
Mm.
That's like spiky bits. So there's no way, like we have yet to scratch the surface of an almost infinite chasm of living things, not
Mm.
to mention the fossil record of those living things. Like
Yeah. Yeah.
yeah, whenever somebody says like they're making a genome LLM, I'm just like, I can't wait to see the garbage you're pulling.
Yeah, hallucinations on that scale are they're going to be
Dear
fun.
God.
Now, we've got to talk about networks and the community that you are a very
Sure.
prominent part about. So, I'm just going to throw that as an open question. Like, how are all of these people doing this kind of work connected to each other? How do new people come into it? Just give me a
Yeah.
snapshot. Like, what are people getting into if they want to join this
Yeah.
very
So
exciting dynamic?
So it was really, really exciting in the days of DIYbio.org, right? So they made this like Google mailing list of like-minded individuals. It was kind of like a forum through email.
Mm.
And that turned into the biohacker movement of sorts, right? From like 2008 and onward, community labs were popping up everywhere, GenSpace being the first one, and then it became in Brooklyn, New York, and then BioCurious and Counterculture Labs in California. And they started popping up everywhere. And iGEM, the International Genetic Engineering Machines Competition, coincided with that kind of interesting renaissance of amateur biology, of amateur genetic engineering, where you have the collegiate folks and then like the weirdo outsiders that have ideas as well. And it was so beautiful. And then like a couple bad actors came in and tried to like co-opt the space as more of bioengineering without regard for regulations, as almost like protest in a way
Hmm.
that became it just became this like edge lord fest that uh, attracted grifters more than anything. And the whole time, like I was there since the very beginning, and or as early on as it be significant enough for me to say that I had a, a long-term presence within that DIY biospace. And I've seen over the years a shift, a change in the guard from folks that are like curious and they want to explore the living world and like make things and do cool stuff, to these like strange longevity grifters that just kind of made everything about making startups and how you can like hack the system to make profit somehow, right? Like the profit people showed up,
Mm.
and things started changing dramatically, and the vibe was off, as the young folks say. Just like there's just the vibe was dead. And I'm hoping that within this AI accelerated, you have access to all the information, albeit hallucinated sometimes, era, we can kind of revitalize that, right? Because currently, there's just, when people say biotech, they think of pharma
Mm.
because they think of profit and money, all right? And like, there's so many things that biotech encompasses that is not pharma. And so there was this we have to do some like unlearning before people can like really come back into it. The folks that I would love to attract are the ones who are like genuinely curious about the living world, want to interface it, want to have make art with it, want to engage with it in a manner the way you do like going to an archery range, just shoot the bow because you like shooting the bow, right?
Mm.
Like that hobby level. Because right now hobbies have turned into accelerated platforms for startup entrepreneurship. And that's really depressing because in that pragmatism comes tunnel vision, right? We're
Yeah.
back to like making product.
Yeah.
And so like all of the cosmos to explore, but we're like, but that one rock has platinum in it. I'm like, you
Mm.
have a solar system to explore. Like, but you're like, but that one platinum rock though, right? That one asteroid. And so yeah, right now is just a really weird time to get into it on a community level. I'm still searching for folks like that. I haven't done a ton of that searching because I'm also a little bit disillusioned by the moniker of biohacker, because that kind of got muddied up with folks who are just like doing publicity stunts, for lack of better words, and
Yeah.
like violating regulations left and right, saying regulations are stupid, and just kind of undoing a lot of good work. I mean, we got to the point where the FBI had an outreach program for DIY bio people, that you would like completely voluntarily have a conversation with the local agent, and they would give you kind of a green light to do whatever you want to do within the law, as
Mm.
long as you tip them off to any bad actors within the space, right? And
Was
that
th
was,
was this an era where there were people like injecting themselves with things and putting out press
yeah,
releases just to get
hmm
loads of
yeah.
eyeballs?
Yeah, it was. And it's unfortunate because it was from one of the most prominent folks within that space,
Mm.
right? And they managed to make like a way in which they can make a business off of the folks by selling kits and whatnot, but obviously violating the CRISPR patent and a lot of Nagoya Protocol stuff by shipping some of those things, and also
Mm.
doing so in like a not hygienic manner. Like they've abandoned
Yeah.
autoclaves. You don't need to be sterile. Yeah,
Mm.
just a lot of just a lot of grift swallowed that community. And after COVID, unfortunately, a lot of the funding for these labs went away because they couldn't be active to generate membership to pay for the
Mm.
rent,
Yeah.
right?
Yeah.
So like they still exist. There's just like murmurings of it, and I don't know what would be the tipping point to get us back into that mode. Um, like God knows I want it to happen. I would love to see more people passionate about this, but right now in this like global economy where everyone's just kind of, will I have a job?
Mm.
It's hard to not feel that anything that isn't towards product or profit or a startup or something is a waste of time, right? And I find that like that's such a like a departure of the human soul. If to just say like I must only do work, anything else is a waste of time is just so heartbreaking. It's like
Mm.
that Churchill quote when they wanted to cut the humanities budget to feed the war machine, right? And Churchill, it's misquoted, but I love this headcanon of Churchill going that what the bloody hell are we fighting this war for, if
Yeah.
not the humanities, right?
Yeah. Yeah.
Like you want to cancel the very thing that keeps us human, right? In order to fight this war against tyrants that want to take our humanity. Um, Not to get romantic, but there is like what I want to get out of my personal practice with biology is it being a craft and a vehicle for me to express myself artistically, but also just this it's a for me it's a like science is a pillar of personhood. Like I find it as just so universal, and we can't just rely on a couple people who are curious to kind of drag us kicking and screaming into the next era, right?
Mm.
And the problems that we face as a species are so complex that there are no more lone wolf geniuses in the woods that can like solve this. It's
Mm.
all collaborative. And currently, right now, because of AI and this hyper competitive space, it's the opposite of collaboration. Like I'd love to see more community biology. And so, one of the like the funnest projects that I was involved with, I don't know if you heard of the Blue Soup saga.
I saw that listed on your website, but I didn't
Yeah.
dig into the full story. Tell me more.
Okay, so um, my dear friend Aline Beckett, who works at UC San Marcos in California, she gets a birthday soup from her mom every year, and she as every year she ate her soup. Her mom's Ukrainian. She has this cool soup, and she forgot it in the fridge after a while, right? And she was like cleaning out her fridge, and she took out the pot to throw it out, and she lifted the lid, and the pot was like cerulean blue. The surface was this opaque primary blue, like as blue as the definition of blue, right?
Mm.
And it was like it was thick blue. It wasn't like the tint of the clear liquid was it. There's just this thick blue everywhere.
Mm.
And she's like, "Oh, that's so gross." And she took a picture and threw it out because she's a microbiologist. And so she posted on Twitter, be like, "Look at this nasty ass soup," right? And as soon as I saw that, I'm just like, "Oh my God, let me sequence it. Like, please send me a
What is
sample."
it?
And so many people were like, please, 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 please go back and get a sample. We want to know what this is. It's
Mm.
so like astoundingly blue.
Mm.
And no part of her soup was blue, right? And so we started this like Twitter thread of like all of the chronically online academics from Twitter kind of bullying Aline into getting going back into the trash and getting a piece of her rotten soup to then like send to me so we could start doing stuff, right? And it became this like stone soup fable where like everyone was offering their resources, like, oh, I have an NMR, let's figure out what the blue is. I can do
Yeah.
sequencing, I could do this. And I'm just like, I don't have any of that, but I could do classical microbiology, right? And so I was trying to identify the microbe by doing a bunch of like classical tests before sequencing and whatnot.
Mm,
And trying my goal would be to recapitulate the blue genes in E. coli to prove that that gene model we have is true, that
hmm.
that's what's causal to the blue.
Yeah.
Yeah, and it turned into this like really beautiful collaboration with so many people. Like once Eileen publishes that article, it's going to have like 50 authors,
Yeah.
and
Yeah.
it culminated in a PopSci article called the soup that blew up Twitter." uh, the IFL Science made it like a short blurb on that and whatnot. It got a decent amount of press,
Mm,
but it's just so fascinating the amount of excitement and amount of like immediate communal camaraderie around this like weird thing, right?
mm, mm,
And so Aline sequenced it and she found that it was a Pseudomonas carnis that was found in a similar species was found in soy milk that turned blue, mozzarella cheese that turned blue. And one of the caveats is that it has to be in a cold environment. It's a
mm,
saccharophile, so it
hmm.
really likes cold. Otherwise, it grows incredibly fast, like terrifyingly fast. It was one of the fastest growing microbes I've ever worked with. Like I struck it out, and in like 3 hours, the plate was swamped. I'm like, what? Like, could not believe my eyes. And it terrified me, right?
Mm,
Now we know
mm.
it's like biosafety level 1, so it's fine. But the blue mechanism is still elusive, right? And so I'm still hunting through that process.
Oh, it
But
needs to be induced by certain conditions to produce the
yeah,
blue.
yeah. And
Yeah.
so and so what she did was she got her mom to replicate the soup, the original soup, and then she sterilized it and keeps it in the freezer in aliquots that are like standardized Ukrainian soup. And it's going to be really fun because in the article, under the methods section, it's going to say Ukrainian national heritage cannot divulge recipe, right? So they'll just give you the objects. Yeah. Because her mom is like, I'm not telling anybody. This is my family secret. And so we'll have the components roughly, but can't
Mm,
tell you like the things. Yeah, so I'm gonna try to like do more media studies to see if I can tease out that blue in a more controlled manner. And because as soon as she inoculates the soup with it and puts it in the fridge, it turns blue, guaranteed.
mm,
But
mm.
no media thus far have made blue as much as that soup does. So
Interesting.
there's
Never
this
mind patents. You need like little Ukrainian grandmothers with trade secrets.
exactly, yeah. And so along those lines, a couple days ago, one of my bottles of a detergent called Triton X100,
Mm,
I use it as the surfactant to help me bleach leaves before I make tissue culture. Because if I take a leaf from the open air, it's full of contaminants and fungi
mm,
and whatnot.
mm,
And so normally you want to bleach it, but my petunia is very hairy, so you need a surfactant to break that surface tension. Otherwise, the bleach can't make contact with the surface.
mm.
And so I use this as fairly expensive detergent, and I had a 10% solution, so that's 100 grams per liter. And out of habit, I always swirl my bottles if there's like condensation on the top, because I don't want the water concentration to affect it if there's like humidity at the top. I want it to be
Mm,
homogeneous.
mhm.
And I swirl it, and immediately, like 20 years of failed tissue culture trauma and contamination just triggered because I saw floaters in my bottle, right? And I'm looking at it and I go, okay, that's contamination. I know a living thing when I see one. That's not
Yeah.
sediment. That's nothing weird.
And how could
Um,
it live under those horrible conditions?
in 10% intense soap, right?
Yeah.
So like 100 gram per liter of soap. So something got in, right? And so I'm just like, okay, you know what? Let's do a blue soup to this. I'm
Mm,
curious. And so I post online. I just sent a video. I'm just like, boo, my Triton stock got contaminated, and it was like a liter's worth. So it's like $100 worth of this surfactant that I only
mm.
use a tiny, tiny bit at a time. And people were like, my condolences. That sucks. Other people were sharing stories of like, oh, something grew in like pure bleach once. I'm like bleach, but bleach. The thing that's supposed to kill the everything, that thing survived in it. Cool. Not only survived but thrived, right? And so people
Mm,
are sharing funny stories. And one person DM'd me and said, "Hey, I'll bankroll the sequencing." It was like
mm,
$120. Like, let's
mm.
sequence it. I
Yeah.
said, "Sure, let's do it." And so I sent it out to Plasmid Source, my favorite DNA sequencing company, because they're amenable to like weird stuff. Like I always send them weird, strange things. But the so I know now that it's in the process of being sequenced, right? So I'm like literally at the edge of my seat trying to know like what on earth could survive in this. And then once I get that sequence and it tells me that it's a microbe, whatever the microbe may be, I'm hoping that it's not something terrible.
Mm,
But if it isn't terrible, I have to culture it. And I only have
mm,
so little of it left because I sent the bulk of it for sequencing.
mhm.
So now I have to figure out a media assay to figure out what does it like to eat, given that it lives in 100% soap, right? Like, do I need to give it soap? Will it die outside of soap, right? Because
Mhm,
if I can
mhm,
maintain that, a lot of academics were saying like, hey, this could be a cool like oil cleanup bug
mm,
because it's eating the soap. It must, because Triton is a long carbon uh, what's a hydrated carbon chain,
mm,
right? Really, really long carbon chain. And so that's food for the intrepid microbe, right? That is food, and life finds a way, just like the Jurassic Park quote.
mm,
And so now I'm like reliving that blue soup vibe.
mm,
And I think if I keep doing this, or if other people continue in this type of vein of just community-based social media research, it could kind of defibrillate that vibe of old science Twitter, where
yeah.
people were just kind of like in a bar talking about science and just having fun before the Trumpians came in and ruined the fun. But that I don't know. I find that to be the most satisfying type of science. This like rapid, informal, like quick, let's do it together, let's collaborate, let's just be excited about something while the
Mhm,
world is on fire, just for a moment, you know? And yeah, with all the technologies around today, you can sequence essentially whatever you want within reason. Like
yeah.
plants are harder to sequence, some can be super polyploid and um, like one of the coolest flowers, the Fritillaria malingensis, the snakehead lily. It
Mm,
has like a checkerboard pattern on its flower. It looks like a tulip, a sad tulip. It's the second largest genome on the planet. It's like 80 gigabases haploid, and it's like a dodecaploid, right?
yeah.
So it's like nine unsequenceable organisms exist. But
Mm,
within microbes, it's a little bit more sane. But there's just so much of this that we can do, right? Like people get contaminations in their labs all the time, and more often than not, they're benign, but you have to kill your media and make it fresh.
mm,
What if we sequence that and kind of make a paper on here is the flora and fauna of the laboratory, right? Common contaminants or things like that. Like you can make it pragmatic, but I also
mm.
just want to do it for the fun of it.
And
Like get
ultimately, that kind of
academics excited.
curiosity, those extremophiles often end up having really interesting genes that become the next CRISPR. They're a tool that can be repurposed in all sorts of unexpected ways.
Yeah, I mean the opportunity cost of not doing so when everything is accessible, especially if you have the privilege of the resources to do so, I find it just like almost in bad taste to not do things like this. Like if I have a
Hmm.
fully functioning laboratory and I have a little bit of spare time, why can't I make biological joy a
Mm,
thing, right?
mm,
And
mm,
just kind of like share that communal experience because if I can be a catalyst for somebody to dive deeper into one of these questions, or they sequence their own thing and they discover something fantastic, wonderful. Like
mm,
I have no grand ambitions of discovering things. If something happens that's cool, awesome. Like I've adopted this like kind of existentialist view of just like it's such a privilege to be alive. Like we're so lucky to be alive right now to do
mm,
stuff that the nirvana of just being able to do that has like superseded any like larger ambitions for fame and glory. Like I don't care. Like I would love if somehow a very serious paper comes out of it, that's great. But if it
mm.
inspires a kid to go into microbiology because they want to do stuff like that too, that's a win. Right?
Yeah.
That's a generational win, you know.
It's the
Eh,
luxury of serendipity.
yeah, yeah, yeah.
Mm,
And like how can we increase the likelihoods of these like dice rolls, right?
mm,
Or where have we not rolled dice in?
mm,
And and also like discourse about biosafety, right? Because I sent it out for sequencing before I cultured it because I don't want to reduce the amount of physical microbe before I know what it is,
mm,
because otherwise you have to culture a whole bunch of it. And what if it's a sporulating pathogen? I don't
mm,
know,
mm,
right? Some
mm,
people were saying that it could be a mycobacterium, and in that genus, one of them is Mycobacterium tuberculosis, right?
mhm, mhm.
And yeah, I like my lungs. My lungs are nice. I don't want to mess those up. But
Hey,
yeah,
we
there's
should probably wrap up. This has been an
sure.
amazing conversation. I didn't realize we were going to go so long, but I've loved every minute. I
Yeah,
have a
super fun.
final fun question I ask
Yeah.
everyone, and everyone does such different answers, and I say this almost every time, but I really want to hear your answer. If you could wave a magic wand and create a new
Yeah.
life form, what would you wish into existence? And you can have
Oh
multiple
man.
answers.
Yeah.
I'm curious to hear where your mind goes with this.
So, a couple of years ago, I had a dream of I was an asteroid miner right? Just like a blue collar worker in an asteroid field, right? And I look, I was like mining something. The sun's on the back on my back, right? And I could see the shadows, and all of a sudden I see this like looming shadow behind me, and I look back, and it's just this enormous celestial vine whose like diameter is larger than the horizon, just moving forward, in wrapped around the asteroid belt, spanning the entire Jovian belt, and it's just like harvesting the nutrients and doing photosynthesis at like the slow geologic cosmic scale time frames. I'm so fascinated by the tenacity of life and how far it can go. And we see papers about microbes surviving re-entry, right, on the
Mm.
surface of space shuttles. Like, how does that even work? I mean,
I,
like 10,000 degree plasma, and this thing is just like, I don't care. I'm in a little crack. It's fine, right?
I think the
And
outside of the ISS has got things living on it as well.
yeah, there's some fungi that penetrated and it's just kind of chilling. And how is that possible? Because water will boil completely in a vacuum, so and all life is aquatic. But my magic wand would be the like space life
Mm.
in space, right? Because
Mm.
biosphere to biosphere, at least we have one example that that's real and that's possible, right? Because our Earth, we have 9 planets, and some argue that there's 8 only. I like Pluto, big fan of it, grew up with it. Pluto's my homie. Um, but if you have a life form, not intelligent life, but just a life form that can survive in the vacuum of space, but work on like geologic timescales, incredibly slow. Its metabolism ticks once every millennia, but it is a living thing and it
Mm.
can evolve. What does evolution like in zero gravity with no atmosphere, getting bombarded by solar death rays, right? Like I would love plants in space, right?
Hmm.
Like this
Hmm.
like strange like lithotrophic like rock eating vines. Like I just that dream I keep having, and I don't understand why I keep having it because normally I don't have repeat dreams, but this one is like different scenarios of me just like with a laser mining a rock, right? Collecting it in the thing, and then just like this looming vine of like unfathomable diameter, right? Really Lovecraftian, but at the same time, the what I find unfortunate about terrestrial life on land is the tyranny of gravity, right?
Mm.
Like we are held down, and such our morphologies are so limited to it being pragmatic, because otherwise we can't move to get food. And I mean, nature's come up with brilliant ways, incredible, and like there's crickets with gears in their legs for
Mm.
added mechanical advantage. I mean, that's insane. But in the deep sea, where you are neutrally buoyant and you're divorced and decoupled from that tyranny of gravity, you have the most like elaborate and beautiful morphologies.
Hmm.
Like if I weren't working on plants, I'd work on jellyfish
Hmm.
because it is just so mind-boggling that a thing that barely has neurons, right? Or it's like super decentralized has is it's just like a sentient plastic bag. It's just this weird gel that does stuff. It's such a minimum viable life form, and there's so much abundance of it. And
Mm.
I don't know, just miraculous designs, like beautiful, beautiful, like fashion, like jellyfish fashion would be something I would get into. Like designing it, but in drapery. Like I would be a tailor rather than a sculptor, right? But imagine
Oh.
that, but in space, like celestial jellyfish creatures. I
Hmm.
think plants are. I go with plants because like there are lithotrophs and there are photosynthetes. And there's plenty of sun, right? Uninterrupted, terribly horrible sun that blasts you with 100%. But I can't imagine that if life is capable of existing on in a biosphere, that it's solely isolated to that biosphere that has to reemerge every time.
Mm.
Like
Mm.
I would love for panspermia to be a thing to some capacity, right? But a lot of the panspermia theories are talking about survival long enough to land on the next biosphere.
Yeah.
I'm really interested in truly independent spacefaring organisms, right?
Hmm.
And
Hmm.
what does that look like? How do you survive gamma radiation? Do you eat gamma radiation? Can there be a celestial vine around the accretion disk of a black hole just nomming at the horizon, right? Like just really cool sci-fi stuff, but I don't want to believe that it's impossible,
Mm.
right? And I
Mm.
feel like the tenacity of life is just so massive that something could survive. Maybe multicellularity is a privilege of a stable biosphere, maybe.
Mm.
Or maybe not, right? Like what if you can have long chains of single cells, right? Cyanobacteria, for example, right? Just make these little mats.
Mm.
Um, the only caveat is that like water exists in ice in space. Rarely do you find it in liquid. And so all life is aquatic that we know of. So unless it's like working in this like sockwood, this like gel state, and it just moves at like an inch per millennia, I don't know how we would survive, but that just excites me. So if I had a magic wand, like space vines for
There you
sure.
go. There you go. I love that answer. I love that answer. So, where can people learn more about your work? Where should we point them to? I'll put links in the show notes.
Yeah, so I have oh, give me one second.
Mm.
Um, I have a open lab notebook that I can either send you a link or just tell you the shorthand for it. It's tinyurl.com/ATGC.
Ah,
FFE.
how did
Yeah.
you get that?
So that's a tiny green cell, flowers for everyone. That's like my main un-PhD thesis that I'm working on.
Mm,
It's just a culmination of all my protocols and my day-to-day musings in like a diary format with a bunch of experiments. So that's like my open lab notebook. That's where all my work lives.
mhm,
And yeah, I'm chronically online on both Twitter, I'll never call it X, on Twitter and on Blue Sky under a tiny green cell on all social media. Yeah,
mhm.
and I'm a walking book, and I'm happy to talk about this stuff because it buys me an excuse to not work on the really heavy stuff for just a little bit and refuel me with these really cool questions.
And I just have to ask, did you come up with the name A Tiny Green Cell because it's ATCG?
So yes and no. I
Oh.
want to take credit saying that it's clever, but it was actually in hindsight that I noticed that.
Okay.
Really, it was just about my real deep obsession with the first protoplast I ever isolated, because I spun it in a centrifuge so hard that all the chloroplasts settled to one side of the dish, and I could see the plasma membrane like an eclipse on the other
Yeah.
side. And that was the
Yeah.
first, it gave me goosebumps because I saw essentially something that is molecules thick, like
Mm,
two phospholipid bilayer thick that I saw with a microscope. And it was the fragility of life was just encapsulated in that one moment, and it gave me severe goosebumps. And
mm.
every single time, I'm just like, okay, that haunts me, right?
Yeah. Yeah.
And it just so happened in hindsight to be ATGC, right? Which
Yeah.
is really cool.
Yeah.
But I have to be honest, I wasn't that clever. Yeah, it happened. I was just like worshiping at the altar of protoplasts after they beat the crap out of me for so long. And it was actually my first protoplast isolation. I literally got one protoplast. Everything else was popped.
Mm,
And so
mm.
I have a kind of like a ghostly photo of a single protoplast just floating in the void. And that was just so, there were so many things that lined up at that moment that just made me feel this is emblematic of everything I do. This
Yeah.
just like incredibly fragile, incredibly precarious little thing that has a tremendous potential, but is in just like a vacuum of nothing, right? It's
Mm,
just like hearkening to a little bit of the melancholy of doing it by yourself in like an environment that is like I live in the sticks now in Huntington, Long Island, which is close to a beach, but not close to anybody I know,
mm.
right? So it's just like this monastic kind of in the canopy of the trees doing genetic engineering in the middle of the night. So is this like loneliness component that I'm embracing as well and part of my art practice. But yeah, it's just like really interesting, stimulating stuff that came out of serendipity. You
Mm.
know, like I'm a
That's
byproduct of luck and enormous amount of perseverance. That's
yeah.
it.
Yeah.
Like it's
That's
not about
that.
intelligence. It's just roll with the punches and get through those failures.
There's
It's
so
worth it.
many lessons that people can take from that, and I'm hoping through this episode and the podcast in general that I'm getting more people excited and inspired to get involved with playing around with biology and also culture. I talk to people who tinker around with culture too. But we'll better wrap it
Cool.
up there. This has been an amazing conversation, and yeah, Sebastian, I am so grateful for the chance to have a conversation with you.
Thank you so much for this opportunity. Yeah, it was you gave me the fuel to keep going now because I have an experiment that's going to run until 3 in the morning. So thank you so much for this very, very much needed distraction and for helping me realign with what I actually love most. Because if I don't say it, I tend to forget.
Hmm.
And so sometimes just forcing it out of me makes me realize just I absolutely love this and I need to do more.
Hmm.
Yeah. So
Brilliant.
thank you so much for the opportunity.
Recombination Nation explores how cultural and biological experimentation can lead us to a post industrial future, one that is more interesting and plausible than either Star Trek or Mad Max. If you want to support my work, sign up to my Substack where you can read free weekly updates from my experimental farm, book reviews, and deep dives. A paid subscription gives you access to the audiobook of Taming the Apocalypse, a bold survey of the hidden domestication potential across the entire tree of life, plus my archive of over 150 past posts.